waking up as we get going today. The handheld mic? Yeah. And so for context, we're going to begin today with a pretty broad definition of what biomimicry is. We'll talk about this again and again. We'll talk about this. Whoa. We'll talk about this again and again throughout the day. But the one I'm starting with here is that biomimicry is the design and production of materials, structures, and systems that are modeled on biological entities or processes. And then as a kind of experiment, I've added for today, or borrowed from biological entities. This kind of broadening that maybe expands the scope of what we're thinking about today. And maybe that's that expansion doesn't work, but I think it's the kind of thing we can begin talking about. I'm going to walk very quickly this morning through some university context for this symposium and for our interest in biomimicry at NC State, a little bit of big thinking about why biomimicry, some discussion of business as usual. If we just keep doing what we're doing, what it's going to look like, a case example of business as usual, and then a call for a more ambitious future around biomimicry and some hope for what we can do together. First, the university context. So I'm coming to you today from the Office of University Interdisciplinary Programs. This is a brand new office that aims to connect things across campus. It was created last year by the provost. I became the interim senior vice provost for interdisciplinary programs in February. I started the job full-time this summer. And so it's a very new effort to amplify interdisciplinarity across campus in education, in research, and engagement with the public. This office has 3 key goals. So one is to provide better interdisciplinary resources to campus in order to lead and administer the institution. And this is going to get less administrative in just a second, but this is some important context, I think. The second is to make it easier to undertake interdisciplinarity across campus and in teaching, training, and research. And the third, and most germane to today, is to create a synergistic team of interdisciplinary leaders that engender change in interdisciplinary teaching, training, and research across the university. And that third goal is achieved in no small part because of all of the amazing programs that are led out of the Office for University Interdisciplinary Programs. And the number of programs is growing and shifting, and as one measure of this, 2 years ago, only one of these program directors was in their current role. All the others have been hired in that time, or hired into their roles. And so it's a really rapidly changing landscape. And it's a landscape that includes all sorts of different kinds of entities that reach across campus and into the community with interdisciplinarity. So we have the Data Science Academy, the Genetics and Genomics Academy, the Global One Health Academy. I'm glad to talk at great length about these. I won't today. We also have the North Carolina Plant Sciences Initiative, which is really a university-wide interdisciplinary initiative to reimagine the future of agriculture. And then we have the Integrative Sciences Initiative. And so this is an initiative to think about how do we take molecular sciences and connect it holistically across disciplines on campus as a way of reimagining the sciences in general, but also to connect it beyond the sciences to how do we connect sociology, how do we connect history, how do we connect the arts to how we're thinking about the molecular future and what's possible in medicine, agriculture, and other fields through molecular innovation. And so this is a really bold and exciting endeavor, partially funded by the legislature, partially funded by the great deal of work Josh does talking to potential donors. And this sets the stage for what we're doing today, because one part of this initiative is to build this amazing new building, an architect's rendering of it here, and this might not be the most recent version, but it's similar to the most recent version, that will be right across from Hill Library. This is a $180 million building through which we hope to begin to integrate molecular sciences. And one of the ways that we'll start to do that in this building is that we will expand our cluster program. So we have a Chancellor's Faculty Excellence Program of cluster hires that includes 20 interdisciplinary clusters, 80 faculty hires, and we're going to add to this at least 2 additional strategic clusters that relate to what we're going to talk about today. And so one of these will be around integrative sciences with a focus on molecular biomimicry. And so this is an opportunity to expand our hires in the space we'll be talking about today, or at least a subset of that space. And then the other is we've just approved another cluster of hires in biomanufacturing. And so you can imagine a kind of connected landscape. So on the one hand, we start with natural historians, evolutionary biologists, ecologists, looking to nature for what's happening in nature, both on the organismal level, on the cellular level, on the molecular level. And then that's then connected to this world of molecular innovation, to making new molecules, to studying how those new molecules can be used and work, and then also in bringing technologies and products that are informed by those molecules or that use those molecules to market through biomanufacturing. And so there's really great potential here. And so as we think about this potential, one of the questions is, why should biomimicry be part of what we're doing in this landscape? Why should it be part of this? And my inclination always in thinking about a question like this is to step way back. And so I'm going to start by thinking about the business as usual scenario for the future of humanity. And if we think about the business as usual scenario that we often see presented for the future of humanity, it is a very technological future from which life has largely been expunged. And so if you look up future cities on the internet, if you look at future human habitats, what you very often see is almost no place for the living world. And I was looking in this one, I think that might be some green life right there. And this is very, very common. It's also the model that we hear about from Elon Musk in interplanetary other worlds. You know, there is some green life somewhere in here, but it's contained inside a bubble. And that's our default model. And this default model has all sorts of assumptions, all sorts of business as usual assumptions that will lead us away from the kind of biomimicry that's possible. But if we confront those assumptions, I think there's the potential to do really exciting things moving forward. And there are lots of assumptions here. I have a short time this morning, so I'm just going to walk through a couple that I think are particularly germane to our discussion. One is the assumption that we know everything. And here the rule is actually the reverse, that we know very little. And for me, the big awakening moment and with regard to this assumption was when I was a graduate student and I became familiar with the work of Terry Irwin, the guy you see here on the screen. Terry Irwin was a beetle biologist. In fact, he wasn't even just a beetle biologist. He focused exclusively on a group of beetles called ground beetles that for reasons of quirky nomenclature only live in the tops of trees, most of the world. And so Terry wanted to collect lots of these beetles so he could name these beetles and figure out how they relate to each other. And so he would fog trees. This was a new method. And when he did, all these beetles would fall down into the ground and he could name them and figure out what they were and begin to study them. But as he started to do that, he realized that almost all the beetles he was finding were new to science. And he brought some of his friends over and they're like, well, all the butterflies are new to science too, and all the moths and all the ants. And another biologist reached out, Peter Raven, and said, Terry, how many species of new insects, if you take the forest you're studying as an example, do you think there might be on Earth? And he said, well, I'm not comfortable doing that, but they thought about it for a while. And he did this kind of bar napkin study where he estimated how many species of arthropods there might be on Earth. And he estimated that there might be as many as 30 million extant species of arthropods. That was a big deal because at this time there were thought to be 1 million species of everything on Earth. And so he basically broke open the possible world. And this even makes its way into poetry. And so this is Jim Harrison writes at one point, sitting on a stump, I feel a little closer to the idea that I'm a member of just one of possibly 30 million species. And so there's this sentiment that the world is sort of cracked open and the possible is much larger. Now, entomologists will proceed to argue about that, whether 30 million is right or 6 million or 8 million. Current estimates suggest that there are probably 7 million arthropod species, 80% undiscovered. But as time has passed, it's become clear that this is not even the big part of the unknown living world. That yes, most arthropod species are not yet named, but as we go into the smaller things crack open even more. This is an amazing photo of a colony of Pseudomonas aeruginosa, one bacteria species. Recently, Ken Losey and Jay Lennon decided to figure out how many bacteria species there might be on Earth in total. And so they used some scaling rules to try to make an estimate, and they estimated that there might be 1 trillion species of bacteria on Earth. And this has been the subject of lots of discussion and argument, but even if they're off by 3 zeros, we get 1 billion species of bacteria. which is a lot, and just 30,000 are named. And so whatever that yawning abyss of unknown bacteria species, it's far bigger than the known. And so take home one in this context is that most of what is discoverable is not yet discovered, and so assume the unknown. And in a biomimicry context, most of what can be mimicked has yet to be seen, much less inspired application. And so for me, that's very exciting. The second assumption about the future that I think bears in our conversation today is the assumption that we're independent. And so if you think about this Calder mobile, I think where we often think we are in the mobile, if each piece of the mobile is a species, we tend to assume we're at the top and the rest of life kind of hangs from us and is primarily just influenced by our actions. Instead, I think the reality is we're one of those bottom pieces. But that's not how we act. When Elon Musk flings people into space, the explicit statements he makes about what is possible and the role of the living world assume that humans are fully in control and fully independent of that living world. Same for Jeff Bezos. I almost felt like I needed to censor that rocket, but that just is. And in reality, we are this piece. And this has become ever more clear in recent years. And what's become clear is that what is remarkable about humanity is that we're like other species in our dependence on the rest of life, but even more so. And in this context, I always think about termites. To orient you, this is a termite queen. She's swollen with eggs. It's a hard life to be a termite queen. Here's the termite king. And in termites, we've known since the late 1800s that termites and their gut microbes are mutually unculturable in many cases. So you can't grow the termites without their microbes, and you can't grow the microbes without the termites. You can grow some of them, but not all of them, and it's still true today. And the reality is, with regard to the rest of life, we're still not mutually culturable. We're still mutually unculturable as well. And this has become really clear in the last few years as we've trained the tools we have for studying microbes in non-human animals and the human animal. And so we now know that a whole suite of our disorders, inflammatory skin problems, ear infections, cavities, inflammatory bowel, Crohn's disease, allergy, asthma, all relate to varying degrees to our failure to be exposed to or to acquire key species from our mothers, from soil, from plants. We depend on these species for our bodies to work properly. We also depend on thousands of species or tens of thousands of species for our food, each of which is dependent on other species. And this is the reality of space travel. This is the reality if we think about the future, which is that if even somebody who imagines themselves to be alone on the International Space Station, in fact, relies on microbes that they acquired from their mother on Earth. They rely on exposures to plant and soil microbes from Earth. All of their food is coming from Earth, and all of their feces is packed back to Earth to be decomposed, not by dung beetles, but by Earth microbes. And so our connectedness and our dependence is extreme. And if we think about the termites, the way the termites deal with this is that they have mechanisms to ensure they pass on their key microbes to the next generation. The queen, and to some extent the king, flies with those microbes to ensure that new colonies have them. And so the termite queen, when she flies, what she has to carry is her microbes. Our trick is we have to carry everything. And we also have to carry the species in which we might someday depend, but we don't yet depend. And so the general take home here is that we need ways to ensure species are safe. We need ways to ensure that we have the right species. We need ways to ensure that they're equitably available. We need ways to ensure they're passed to individuals in the ways that they're able to establish. We need to do all this while being responsive to the different needs of different humans, cultures, lineages, individuals. Huge challenges here, but also opportunities. But in a biomimicry context, for a successful future, we need to far more rapidly find ways to learn from nature and to apply what we've learned and to save those species so we can continue learning from them. And we talk about this potential, but we haven't fully realized it yet. And then assumption 3, and this is the last one, I could talk about these assumptions for a long time, is that we know ever more about life. And this is an easy assumption to make. It's sort of this assumption of progress. And it's true collectively as scientists. Our collective body of work is greater and greater. But if we think about what we individually know about the biological world, there's a fair amount of evidence that it's becoming less and less. And I'll provide two contexts for this assertion. The first is, if we think about what the average child knew in a traditional society 12,000 years ago, the average child would be learning thousands of species around them. And we see hints of this if we look at indigenous people still living relatively traditional lifestyles. And so, for example, I worked in Bolivia for many years, and some of the indigenous children in Bolivia can distinguish 80 species of ants. And so there's part of our context for what an individual might know at one point. But the other piece of this is the question is that knowledge still around? Are some individuals still learning more about the natural world? But there's this topography. And so Stephanie Shetler did a study a number of years ago where she was trying to test the hypothesis that rural kids in North Carolina would know more about the species around them than would urban kids. Other hypotheses here, but that was a key one. And so she just had, among other things, had kids in rural environments, urban environments in North Carolina just make lists of species that they thought of, that they liked or didn't like. And what people had seen from other studies is that that's a measure of what you're aware of, like what's most apparent in your environment. And so what Stephanie predicted is that the rural kids would know more about the species in their environment. That's not what she found. Instead, almost all of the kids listed exclusively things they could have seen on TV. And so this daily environment of a child, of a future generation of students, is more disconnected from the living world. But it's also true that at institutions in general, at the academic level, that we've emphasized organismal biology less. And so we're less likely to hire an ornithologist, we're less likely to hire a microbiologist who knows how to culture microbes. At NC State, we have an opportunity because we still have many of those people, but collectively, institutions, we have fewer and fewer of those people, fewer and fewer of those gatekeepers who are trained in knowing about lots of species. People like Terry Irwin, he died quite recently, but when he was alive, he could tell you about 1,000 different ground beetle species and each lovely thing about each one of them. Now, from a biomimicry perspective, being able to connect to somebody like that becomes very important. And to summarize here, to a first approximation, no species have yet been studied. If we round off, we haven't even started. Even for well-studied taxa, most of what's knowable is not yet known. Our persistence depends on our ability to find ways to better learn from the living world and save that world that we might learn more in the future. Individually, we're coming to know less and less about the living world as a result of gatekeepers to the living world. The ecologists, evolutionary biologists, and naturalists will become ever more important as conduits, as models for biomimicry. And to realize this potential, we must more broadly connect organismal biologists, engineers, chemists, experts in design, and other scholars. And so that's one of the reasons it's so exciting that you all gathered here today, because just from the folks I know in the audience, we have at least 10 different departments here from NC State with very different perspectives on this question. And one of the things we'll see today is not all of the talks that you'll see will be intelligible to everybody in a full sense. And so one of the questions we will have is how do we work together to do things we couldn't do on our own, even when our day-to-day work is not fully intelligible to each other. And so what are those connections going to look like? And I'm very quickly going to talk about what this will look like as sort of a business as usual scenario. What it will look like if we don't reimagine something bolder. And I'm going to use a quick anecdote here from work that Adam Taranto here at NC State led in thinking about the future of urbanization. And so Adam developed this model of urbanization, and what you see here in the red is the future urbanization in the Southeast predicted by this model. And the yellow is current urbanization. And what it yields is this giant Charlanta city that stretches in the Southeast. This was really interesting. Adam's now working on a new model. But one of the things that was intriguing about this is essentially Charlanta, in the business as usual scenario, if we keep doing what we're doing, come on in, folks, is that it basically just traces I-85. And so the future of urbanization is predicated on inertia, on business as usual, on where this road happens to be. But what's even more surprising about this is that the reason that I-85 is here is that that's where the pre-colonial Native American path went north-south in North Carolina, South Carolina, Virginia, and Georgia. And it went north-south there because that was the easiest place to cross the rivers on foot. So our future urbanization model is predicated on where it was easy to cross rivers on foot. That might be the best place for a giant city, but probably not. And what's even more interesting is that if we look at Clovis points from 11,000 years ago, from the Clovis peoples living in North Carolina, South Carolina, and the region, this appears to be a footpath. So each one of these is a Clovis point. Most of them are along rivers. This, archaeologists have argued, is a footpath moving north and south. It's in the same place. And so we may be building our future on a 10,000-year-old footpath. And so as we think about biomimicry, we need to avoid just doing the status quo. We need to think about what is more ambitious than inertia. What is a kind of possible future for biomimicry that is active and involves agency and in which we take control of that future. And I'll say that the biomimicry business as usual story, and Clint, I think, will talk a little bit more about this, is one in which ecologists and evolutionary biologists who make amazing observations aren't necessarily particularly interested in their usefulness. This is how I was trained. I was trained that to be useful was a bad thing, and that ecologists should not do it. And there's a lot of culture there, and there's a reason that the field developed that way, but it's how it developed. The engineers and designers that identify new models for biomimicry often do so through reading papers by biologists without direct contact with the biologists. That chemists rely on generalized models of molecules or processes without insights from the diversity of other molecules or processes. And that really exciting artists who make connections between biology and design often have no conduit for the application for those connections. Now, there are amazing people in this room who've done things that are very much not the business as usual. But how do we imagine a community working together in a way that emphasizes a new model? And I'll just give you one quick example of what the business as usual model misses for us. And it's the example that seems far-fetched, but or sort of far removed. And it's an example from the history of the human heart. I wrote a book about hearts a number of years ago. And sort of inadvertently in the book, again and again, I ended up telling stories of where biomimicry would have been useful, but it wasn't. And this is one. So this is the first heart transplant recipient. And if you go back and read the news stories about this moment, it's heralded as the future of medicine. Christian Barnard, who did this surgery, went on to date movie stars. He was in the front of time. He was heroic. But what's not mentioned as often is that the patient died 18 days later. And for the first almost 10 years, so too did every other recipient. And the reason was nobody could battle the rejection of the heart by the body. And what made this possible was the discovery of cyclosporine. And cyclosporine was discovered from soil microbes by trial and error, looking again and again just randomly at soil microbes for what might produce compounds that were useful. Finally, one was found, and then heart transplantation began to save many, many people. And so the secret was cyclosporine. Cyclosporine then inspired new synthetic compounds that do some of the same things to calm the immune system. But nobody ever really considered, like, well, why does this compound do this? Like, what's a function in nature? Until Kathy Hodge at Cornell University was going through a fungus collection with her students, and she found this fungus that she was able to then grow up and identify. And it turned out that this fungus was the same fungus that was discovered to make cyclosporine. And so she started to look at this in more detail, and this turns out and naming fungi and identifying the different life stages is very tricky. This is a fungus that invades the bodies of beetle larvae that crawl into feces because the fungus wants to be in the feces and grow. And so it takes over the beetle larvae's brains and their immune system. It basically keeps them alive, but barely so, and calms down the immune system, and then it grows. And what Kathy has argued is what was happening here is that the reason cyclosporine is working in humans is that actually the fungus that produced it evolved to produce cyclosporine so as to calm the immune system of the insects it was taking over to live out its strange little life in dung. Now, what's exciting here, so what's demoralizing is that had we looked originally for places you might see a fungus like this, we might have been able to discover it more quickly. What's even more demoralizing is since this discovery, there's been very little work to look at the thousands of other cordyceps and related fungal species for other related compounds. And so even though we learned about this potential for biomimicry, we've not followed up on it. And so this to me is business as usual. And there are many, many examples of this. And so how do we leverage our collective potential to do something different, to embark on a more ambitious future? And I think one of the key pieces is figuring out how to work across our disciplines in ways that we're rewarded for this work, we're connected, we overcome the differences in jargon between our disciplines, we overcome the differences in culture, what we value, and we find ways to build conduits. And I think, and this is from some of my own work, that this is actually a really exciting place to do this, not only because we have a lot of the commitment to doing this, but also, as it turns out, NC State is arguably the most diverse, or one of the most diverse campuses biologically in North America. And my little window into this is that I had an amazing student a number of years ago, Benoit Guinard, who studied all the ants on campus. And he found 89 species of ants on campus, which is more species of ants than there are in all of Northern Europe. And what's interesting about this, even if you really don't like ants, is that Benoit later went on to show that if you know how many ants there are in a place, you can predict how many vertebrates are in the place, how many plants there are in a place. And so that diversity of ants, we're the most diverse campus with regard to ants, except maybe University of Florida, is predictive of a diversity of species more generally that we can learn from not far away, but right here where we are. In addition, here we still have organismal biologists. We're investing in molecular biomimicry. We're the best textile program in the country. We're expanding our engineering programs, and we're investing in connecting efforts across disciplines. It feels to me like an exciting moment. And it feels to me like a moment that if we plan now, we can leverage this building that's going to be finished in 2027, using biomimicry to create new molecules in the first set of labs, using new technology to visualize those molecules, creating novel uses of those molecules, connecting those molecules to diverse forms of innovation, but doing so in a way that's connected to natural history. And so the hope here, and I'll close right here, is to work together to reimagine a more ambitious, connected biomimicry that builds on the work of ecologists and evolutionary biologists, draws on many models rather than a few, connects to many societal needs, and fast-tracks discoveries that transform to society. And to do so, we'll have to transcend disciplines. And the good news is that we have a context in which we can be ambitious, and we have all of you amazing people who've gathered, and come on in the door, amazing people. And with you, the potential to work together, not only today, but in the future, I think is extraordinary. And so what we're going to do today is we're going to have a series of slightly longer talks in the first couple of hours. We'll have a little break. We're going to have some more rapid talks. And the idea is to get a sense of the landscape, get a sense of the possible. The early talks are going to emphasize big picture things, and then we're going to dial in on some case examples. And this, we hope, is really the first of many conversations about this topic. And so we want to hear your feedback. We want to hear your ideas. And then the afternoon, we're going to break into a working group of just the speakers to think about how do we write a paper about what's possible at the intersection of these disciplines, and how do we also think together strategically about what it's going to look like to be really bold in this arena. And so I will stop right there. Thank you so much for joining us, and I'm going to quickly tip to our first speaker, and I'm going to remind myself of the order now. And so everybody take that moment to come on in while I'm getting oriented. So our first speaker today is Clint Panick, and Clint comes from Kennesaw State University. He joined us for a while here at NC State as a postdoc. And Clint has really been instrumental in innovating in the space of connecting biologists to engineers, to people in design, to folks in chemistry. And he started this before he came to NC State at Arizona State University. He went and worked with the Biomimicry group again at Arizona State after he left NC State. And now he's really been a pioneer at Kennesaw State University. And so Clint's going to provide us some of the broader perspective on biomimicry as a field. What is the field doing? What's possible? What is his lab doing? And so I'm going to jump back into the audience because I'm super excited to see what Clint has to say. So thank you so much, Clint.
Thank you. All right. Thank you for having me. Oh, I'll go back. So today I'm going to be talking about biologists and biodiversity in biomimicry. It's a lot of bios. And you would think that biomimicry has bio in the name, so obviously there should be a lot of biologists working in the field. But as I learned when I joined the Biomimicry Center, I started looking around. This was at Arizona State. And there were architects in the Biomimicry Center, engineers, industrial designers, visual communication designers, a science writer, education specialist, but I was the only research biologist who was in this center. And actually connecting with other biologists on campus was our most difficult job. When we opened our doors, engineers came in, they wanted to look at things, get ideas. We had architects come in, all these different people, but biologists were hard to connect to. And in fact, this is where the biology buildings are on Arizona State campus, and where they chose to put the Biomimicry Center is way up in the northwest corner. It's actually inside the architecture building. And this isn't something that's unique to Arizona State. This is something that's common to the field. So some research a number of years ago looked at the number of papers published with biomimicry or bio-inspired design in the title. One thing you can see is that the field was rapidly accelerating, so this is becoming popular. But the other thing that the authors noticed was that the physical sciences, chemistry, material science, engineering, were publishing way more papers than people who were in traditional biology fields. And when they looked at where authors were actually based within their departments at universities, they found that only 8% were in a biology department. So biologists are actually a relatively rare species when you look at biomimicry. In some ways, this makes sense because you have people coming from engineering who are bringing new tools, new ways of looking at things that biologists have looked at for a long time. But I would argue that we're missing something when we don't have biologists in the room. And part of that is what Rob mentioned, which is biodiversity. This is something that as a biologist, this is what I get excited about. This is what I'm trained to study. But it's something that can be hard to tap into if you're outside of these fields. And so a study that came out a couple years ago looked at the number of species that had been looked at around some key questions in bio-inspired design. So one is drag reduction. You can see that 30% of studies on drag reduction in bio-inspired design look at sharks. And of those, about half look at only one species of shark, the mako shark. The same happens if you look at adhesion. So if you look at adhesion, the gecko, which is sort of the mascot of this symposium, the gecko's foot was what was on the first slide. Close to over 40% looked specifically at reptiles and amphibians. And of those, 100% were one gecko species. So we're not tapping into the large diversity of things that we have here. The other thing that I want to point out is that if you look at which one of these studies looked at more than one species in a single study, it's less than 10%. So very few people are looking at more than one species at a time. Again, there's some benefits to this. It's like a model species approach. I think that's common to all the molecular biologists in the room. But I think we are missing something by only focusing on one species at a time and limiting our search to just a few of the sort of standout examples. So to talk about, or to maybe make an argument for why biologists like me should be involved in biomimicry, I'm going to go through a case study of some work that we've been doing in my lab on probably the most overstudied thing of all in biomimicry, the honeycomb. And so my work began on this. So some quick background is that I am trained as a social insect biologist. I work on ants and I work on bees and things like that. And so while I was in the Biomimicry Center, we had this engineer, Dhruv Bhatte, who came into the center to see if there were opportunities to do some collaboration. And his specialty was 3D printing honeycomb. And when he came to the center and talked to me about it, in my mind I was thinking, I don't think we can figure out anything new about honeycomb. I mean, people have literally been coming up with models and thinking about honeycomb for 2,000 years. There's a paper from ancient Romans who were studying honeycomb. But I thought, sure, I want to bring people into the center and talk to them. So we came up with a plan. And what it turned out was that Dhruv had never seen honeycomb made by bees. His entire work had focused on honeycomb, 3D printing honeycomb, but he hadn't actually seen real honeycomb. So I said, okay, I'll come down to your lab. I'll bring some honeycomb and we'll go from there. And so in Dhruv's lab, he had a bunch of things laying around that looked like this. Dhruv's specialty is that he 3D printed in metals. So they had this giant room-sized 3D printer that made kind of actually relatively small parts, but they were metals. And when you look at them, they're very perfect, regular honeycomb, very sharp corners, rigid, something that you would expect to see in an engineering lab. When I brought the bee honeycomb, you can start to see that there are some differences. And the big one that jumped out to us, so one thing is, honeycomb is still pretty regular through most of the comb. Bees are really good at doing this. But what Dhruv noticed, he said, oh, whenever they build comb, their corners aren't sharp. they have a little rounded spot in there. And so we gave that a name. We call it corner radius. That's what we measure to study it. And so we thought, what could we do? Is there something, is it maybe that, one idea is that bees are just not great at building honeycomb. If they, had a 3D printer, maybe they would make perfect honeycomb like Dhruv was doing. Or maybe this had some function. And so to answer this question, Dhruv went and then 3D printed a series of comb panels. And then put them on a hydraulic press and crushed them to test to see if they were stronger than the other. This is what this looks like. So it's very sharp ones up in the upper left, and then all the way down to a fully rounded, basically a perfect circle inside the comb. I just like this video. You can't really learn anything from it, but it's fun to watch them get crushed. Okay. And so the output from research like this is you get these things called stress strain curves. I'm not going to go into a ton of detail on this, but this is, when we talk about blending disciplines, this is where it gets hard. I had never seen a stress strain curve in my life until I saw this. And it took me, I think like maybe 4 months of collaborating with Dhruv before I finally figured out what these are. So I'm going to try to condense it for people who also might not have taken mechanical engineering anytime recently. For us, we have the 4 different honeycomb types, one that goes from sharp corners, and then the one at the top is the one that's rounded. From the bee side of things, we don't care about most of this graph. We care about really the beginning down at the bottom here. And so how steep that line tells you how stiff the material is. And so what we see is that adding that corner rounding increases stiffness. And this is something that engineers are interested in. And then where that line starts to round over and then go into the next section, that's where it breaks. That's the failure point. And so if you're a bee, if your comb breaks, like that's the end of the story. You don't want that to happen. And what we find is that adding a little bit of that material in there prevents them from breaking. And this was pretty exciting. So when we discovered this, we were like, okay, right here, I think we have an idea for something that could improve how people are using honeycomb. So traditionally, if you wanted to increase stiffness and you were working with comb, what you would do was just make it thicker and thicker. Or you could also choose a different material. You could use steel instead of aluminum or something like that. But all of that adds weight and it adds material. What we found is that to achieve the same property, you could just add a little bit of material into the corners. You're still adding material, but you're adding much less. So this allows you to build lighter weight parts and also be arguably more sustainable because you're using less material. So with this initial discovery we made in his lab, we submitted a proposal to NASA and they actually funded us. And you can tell that NASA loves honeycomb. This is the James Webb Telescope. It doesn't have rounded corners, but that's for a different purpose. But the thing I'm not showing here is that basically every rocket is wrapped in what they call honeycomb sandwich panels. And I flew on an airplane to be here today. And that airplane, the entire fuselage is wrapped in honeycomb. You can't see it, but it's just under that outside. The wings are wrapped in honeycomb, and even that little door you lift up to put your overhead luggage above your seat, that has a honeycomb sandwich panel in it. So honeycomb is used extremely commonly through aerospace design, particularly because its strength to weight ratio. It's very lightweight and is pretty strong. So there's a lot of applications for this. So with funding from NASA, what our team sought to do, we worked with a private company. So this is a private-public partnership. And the private company was working on building a model to optimize what would be the optimum parameters for honeycomb that you would put in these for any application. And this is adjusting the size of the cell, adjusting the thickness of the walls, and then adding that corner rounding component into it. And so this is what we were interested in studying. We said, okay, this little corner rounding helps, but how much do we need to put in to see a big difference? And so I have already simplified the axes of this graph. Again, this has a lot of engineering stuff that's very technical. These are a little bit more complicated than what I put here. But this is a graph that what it shows is variation in how thick the walls are and then how much of that corner rounding is in there and what the effect is on stiffness. And so what you can see on this end here is that the thicker walls are always stiffer. And this isn't a surprise. We knew this before. So, if you want something stiffer, you make the walls thicker. But what we found also is that when you add the corner rounding, it's the same thing. So you can go from a sharp corner to a little bit of corner rounding to a little more and then to a perfect circle. And the perfect circle is always more stiff. And this is controlling for weight. So it's saying if you want to optimize and build the absolute lightest part, you need to make a perfect circle honeycomb. And this was first of all, I guess it's a useful discovery. So we discovered something, but it kind of was disappointing because for one, the bees don't seem to build perfect circles. And also there's no thing that this company could sell. There is no optimization tool. You just make it a perfect circle all the time and you don't have to do anything crazy. And so while this was going on, this is what happens when you have a biologist on the team. So first, I was able to show them honeycomb when we came up with this idea. Second, there was enough money that was funded through NASA that I got to go off and do my biology things, and that was look at other species, not just honeybees. And so what we use is this Keant system here, and we would put comb underneath it, and we would look at individual cells, and we would measure all those traits, how big they were, how thick the walls were, how much corner rounding was in there. And we didn't do this only on honeybees, but we actually traveled to New York City, where the American Museum of Natural History has the largest and most diverse collection of honeycomb nests in the world. So we went in the back of this museum and then took any nest that could fit under this machine, and we did the same measurements. And so we ended up getting about 70 different species that build in all kinds of different materials that include wax like bees, but also paper and mud and combinations of paper and mud. And then we looked at what the traits were across these species. And so this is what we found from our work. So I just have this plotted, the cell diameter, the size of the cell, and then how much corner rounding we see. And then that dotted line shows you if they were doing what the engineers said they should be doing, if they care about stiffness, they should be following that line. And what we find instead is that they're all under that line. So first of all, they're not doing the same thing. And we also saw some variation here and that we were trying to explain. And so we saw variation between the different materials the bees build. So if they build with wax or mud, they tend to be much closer to that, that perfect round line. And then it also changes as the size of the cell changed. We saw that they were putting less and less in the corners. And so when I came back with this to the engineers, we all sat in a room and kind of were scratching our heads. We're like, how can we write a paper about this? It doesn't really match the engineering model, and what might be going on. And then one of the postdocs in the room said, well, I mean, stiffness isn't the only thing we can measure. We can look at other things too. And so they went back to the drawing board. So they had originally done research on stiffness. They decided to look at stress. And stress is where the honeycomb actually fails. These are the stress points. And if you zoom into the bottom of this, this is from the model we created. If you look at a sharp corner on the left, all the stress is directed right at that sharp point. And that's where the first crack is going to happen, and then the whole thing crushes after that. But if you add the rounding, what you see is it diffuses that stress down to the walls, and this is probably what's giving it stronger stiffness. And we wanted to see if there was a way to not only optimize for stiffness, but to look at stress. And so we did the same kind of study, but here you can see that the plane looks a little bit differently, and the story isn't so simple. So now it's not always build a perfect circle. The lowest stress point here follows this little line about in this part of the screen. And so what we found is that adding corner rounding, the benefit changes depending if you have relatively thin walls or if you have relatively thick walls. So if you look at a thin-walled honeycomb, if you add that corner rounding, what it does is it actually pulls that stress point to the thin part of the wall in the middle, and then it snaps right there. That's where you get a crack point, and it actually reduces how strong and how much weight that the honeycomb can withstand. But as you move towards thicker and thicker walls, then you see the benefit of this. And then when we look back at the bees, I'm not showing this now, we found out that if we change that x-axis from the cell diameter, but instead look at the relative thickness, the bees do the exact same thing. Bees and wasp, I should say, because we're looking at wasp too, which is pretty phenomenal. And this is something, I think maybe like Rob said, 10 years down the line, the engineers could have figured this out themselves. They didn't necessarily need a biologist on the team to figure this out. But this is something that we figured out within, 6 months of getting our funding, partly because we had an interdisciplinary team that was working on this and looking at different things. So I think that sometimes this is, I have other cases where there's sort of more direct involvement from a biologist, but I think there's all kinds of different things and emergent things that happen when you put different groups of people in these types of teams. And so the engineering firm with this was able to create something that did optimize honeycomb. They created a product they can sell. It goes into this program called Ansys, and it's a program engineers use to build anything from machine parts to bridges. And so this is a pretty cool thing that came out of this. And then we were also able to get follow-up funding the next year from NASA to not just look at honeycomb, but look at all kinds of things. And so in addition to honeycomb, we started looking at cactus skeletons, these things called corals, and then glass sponges at the top. And we just filed a patent on glass sponges that came out of this work. And I think this is another case where you look at biodiversity. Part of my role in the team was saying, okay, where do other types of honeycomb materials exist? So I worked with a graduate student and we made this giant list, and we looked everywhere we could in nature to find these things. And then we down selected these, and each of these now have projects going on with them, and each of them has shown something new about how we can improve structural integrity of parts. So yeah, that's my argument, I think, for why biologists should be involved, and biodiversity as well. And I think we're missing it in a lot of the projects that we do. All right, so with that, I'll thank my funders and take any questions if there's time.
We do have time for some questions. I can run this microphone out to anybody who has one. Sure.
Thank you. That was really interesting. At the beginning, you mentioned that a lot of these studies have used just a single organism. And I'm wondering if you think that always necessarily reflects a weakness or a shortcoming in the experimental design, or if it might, in some cases, be that that organism is just a really good model organism that reflects common traits across a larger taxonomic group.
Yeah, that's a great question, and I agree with you. It's basically a model system approach, right? And so I don't think that, obviously we have a lot of benefit from that. But I think that also when we have those, it does help to have someone then zoom out and say, like, what else is going on? There's a great example with geckos. So they focus mainly on one gecko, and then some biologists who were not working in biomimicry published a phylogeny and that looked at these traits and how they evolve across the group. And people, I think, are yet to investigate what they found, but there's a huge variation in the spatulae on the toes of these things. And some of them live in desert environments, and some of them live in aquatic environments, and some of them live in forests. And there's an idea that if you want to optimize for adhesion to these different surfaces, now you have a library to look at. The other cool thing that happened as part of some of that work is that people looked at not geckos, but they looked at anole lizards, which are like the common green lizards that are running around campus here. And they found that they're not as sticky as geckos, so they're maybe not a champion adapter, but they do have spatulae and they are sticky. And the pads on their toes are easier to manufacture. And so that's been the huge holdup with gecko research, is, you know, and why you can't go to Walmart and buy gecko tape is because you can't make this for any kind of reasonable price. And so looking at some of these other things now is opening up opportunities. So I think there is still, like, I would agree that there's benefit in zooming in on a model and really understanding the system, but you also can get benefit by looking. So I think they should be paired together. Well, I think it will be beneficial to pair them together. Yeah.
Yeah, my question was related to energy in the different thicknesses of the wall versus the curvature. And so, you know, biological systems are often optimized for efficiency for maximum reproductive output, but physical and chemical systems are not.
Mm-hmm.
And so just, you know, you make the walls thicker and that reduces whatever it was, stress or the other one. But where is the optimization there in terms of energy that's put into it?
Yeah, that's a great question. And yeah, that I skipped over. So from the bees' perspective, they want to use the least amount of wax as possible because it takes energy to make it. And so to make 1 pound of wax, it roughly takes the digestion of 10 pounds of honey. And so bees are they're very sustainable when they build their comb. They try to use the least amount of material possible. And they'll even use old comb from other colonies, even if there might be disease. And so it seems like there's a lot of selection pressure on bees in particular. And wasps have a similar thing. They don't produce the paper themselves, but from their bodies, like metabolically, but they have to make trips and they chew it up and they mix it with saliva. So there is a lot of energy invested. So at least from their perspective, it reduces the energy if they're able to make these little adaptations, like add a little bit of rounding, if it means that they're using less of the material. And they're probably optimizing to use the absolute least, close to the absolute least amount of material to do the job. Yeah.
Thank you so much, Clint. Amazing. So next we have Josh Pierce, and Josh is in the context of today, is the director of the Integrative Sciences Building and Initiative. And Josh is going to give us a more chemical perspective on the possible. And so be thinking as you hear from Josh about where the intersection points are for these different approaches to thinking about biomimicry. Thank you, Josh. Thanks, Clint.
Thanks, Rob. And yeah, we're all in for something a bit different, but I hope highly relatable to everyone. And it's my pleasure to be here. I think Rob's introduction to this topic was phenomenal. I think the cyclosporine example is exactly in the wheelhouse of certainly what I think a lot about. And so I hope to explain to you a little bit about what I mean, at least when I think about biomimicry. And we'll talk not really so much about our own group's work, but more about just the opportunities that we might have. And so, you know, the heart of all of this, and I think the heart of the Integrative Sciences Initiative more broadly, is to think about how can we learn from nature and from, you know, the molecules in nature to really address grand challenges that society is facing. You know, we have a lot of interest in health. We have a lot of interest in the environment. You know, that leads to food and energy and water. And you know, these challenges need these interdisciplinary teams to solve them. But I think, you know, at the heart of all of the solutions is this sort of molecular level discussion. And so molecules have literally changed our world. I mean, in the most literal sense, molecules make up our world, right? But molecules that we can use and deliver, whether they be drugs or whether they be materials, have allowed humans to have, you know, dramatically increased lifespans, to have, you know, technologies and lifestyles that simply would not be possible otherwise. And if you're interested, there's this book. It's very much geared towards the public. It's called Napoleon's Buttons. And the origin of this actually is that Napoleon, his army, you know, they sent about 500,000 troops to Russia. And they lost about 490,000 of those troops in the Russian winter. And it's thought one of the biggest reasons that occurred is that the buttons that were on the uniforms of the French army were made of tin. But the people that made those buttons didn't realize that tin has several allotropes, and so different types of bonding of those tin atoms, one of which is very, very brittle in cold weather. And so the buttons literally just fell apart on all of the French uniforms, and they had essentially no clothes in the middle of the Russian winter. And so, you know, those kind of molecules are quite simple, but more complicated molecules, the drugs we take, aspirin, amoxicillin is over here beside me, a synthetic version of penicillin, very similar origin story to the cyclosporine that was mentioned earlier. But all of these molecules have really allowed us to live a different life than we would have otherwise lived. And the reality is, when you look at molecules and you look at the atoms that make them up, changing one of them. Everybody's probably seen a periodic table in their life. There's all those atoms that, you know, just have the little two-letter abbreviations. Changing one of those in a molecule can literally change the complete properties of that molecule and ultimately change the course of history because of the effects that molecule might have. It's really quite wild to think about, but it's inspiring as a chemist. And relevant to this conversation today is that nature is a powerhouse of making molecules, right? So right now, outside, well, within us certainly as well, and all the microbes in us, but outside in the trees and plants and grasses, bacteria in the soil, these biological organisms are making countless molecules that have all kinds of different functions. Fungi are making cyclosporine, and bacteria are making antibiotics. They're making bark in trees. They're making Taxol that you know is a very frontline treatment for many human cancers, right? So tree bark is curing human cancer, right? Think about that for a while. How is that even a thing, right? And to Rob's point earlier, you know, that we've only scratched the surface on the type of organisms and the nature of what they are able to do. And so these are chemical structures, and I don't expect anybody to really appreciate what they are except for the chemists in the room. But the bottom line is that nature takes small molecules. These are amino acids. These are little tiny building blocks that are available to it, and it can stitch them together and build up these compounds. All you need to really recognize is that you're connecting these into more complicated things, and that's what chemists do. That art of doing that is called synthesis. It's an interesting term because, you know, you can also synthesize ideas and concepts. And so we synthesize molecules, and that's this process here. And so how do we do that? How does nature do that? Can we learn from nature, and can we mimic what nature does in that process? And that's part of what we'll get at here. And so mimicking a natural process or product at the molecular level is really what I think about when I think about molecular biomimicry, and really with the broadest intent on that term. And so, bioinspiration, biomimicry, these things have a lot of overlap. In the context of my view of this, there's a lot of one and the same in this. But I think there's a vast landscape of opportunities here. And so that opportunity is in synthetic chemistry, as I'll sort of give you a brief overview of today, in synthetic biology, which my colleague Gavin will talk about in a little bit, biomedicine, you'll hear about some engineering approaches to these solutions, data storage and thinking about how to mimic the way nature can store data, so many different topics that are important with respect to how nature can inspire the science and technology and the chemistry of biomimicry. And so one of the ways that we can do that is by mimicking the actual form of nature, right? And so nature, via its many enzymes, and Tom Mackers is here, and we'll talk about some of this chemistry in one of the lightning talks in terms of what enzymatic systems might be able to do for us. But all of those biological systems, right now within us, we're full of these proteins which are able to do chemistry, right? So our body right now is conducting many reactions using these enzymatic systems, these proteins, to do chemistry. So instead of me over in Dabney Hall, these enzymes in our bodies are doing fascinating organic chemistry. If anybody's ever taken sophomore organic chemistry in college, right, all those reactions you learn, these enzymes do many of those reactions right inside of us, and every living organism does it. And bacteria are particularly good at doing chemistry, right? And so all the different bacteria that are known, the 30,000 that Rob mentioned that are named, and the many more that are not, they all have these systems in them, and they all do really powerful chemistry. And the thing is, when you get new organisms, sometimes you find new capabilities that those organisms have, and not only new chemistry that they can conduct, but new products that they make. And those products can be really useful for us. Those can be our next drugs. They can be our next materials, right? They can be the next coatings on your smartphone that can, make the screen scratch less, or whatever it may be, right? There's so many different uses for the compounds you might make, but it centers on learning and being inspired from what nature does with these natural systems. And so here's a couple examples of the structures of the kinds of molecules. Rob mentioned cyclosporine in his talk. You're familiar with many natural products. You've ingested many natural products, right? Vitamins, pharmaceuticals. Everybody's taken these things, right? They're all molecules that have structures that are a connection of these different atoms, right? And so every one of those we could draw in this kind of a format, and we can understand their precise structure at that level. And so this is the atomic level. And so when we talk about biomimicry, we're ranging from thinking about atoms to thinking about honeycombs to thinking about ecosystems and everywhere in between, right? And so I'm clearly at the bottom of that pyramid, but regardless, that's what we're dealing with. And so these enzymes, these proteins in nature, do chemistry to take simple molecules and make them more complex. And so there's a huge opportunity for people to find new organisms. There's a huge opportunity for people to understand those organisms. There's a huge opportunity for people to be able to, in a laboratory, harness the power of these organisms to do chemistry, right? And so we'll hear some of that today, very much literally leveraging the biology itself to do cool things and to understand how the biology does this, right? We can't mimic it if we don't understand how it works, right? And so if this is just a black box, this is a protein that does stuff, and we have no idea how it does it, we're never going to be able to mimic that. And so it's really important that we have folks that are really deeply understanding how these systems are able to do chemistry, right? And so to that end, we can mimic that both literally by harnessing the enzymes, but also by mimicking how those enzymes work now in a completely synthetic way. And so there's a field called catalysis, which is basically using chemistry to promote the assembly of things into more complex things, we'll say, right? So that's a simple way of putting it. And so in this paper, they've developed these metals, so these are again on the periodic table, our fun little abbreviations, right? We have gold and scandium here. These metals, which you can see up here what they look like in the real world, are able to promote that same kind of reactivity that enzyme could do, but now without the enzyme. Right? And so we can just use these metals to do that same kind of thing. And now we can make a vast library of these compounds and start looking for whether they have the kind of biological activity or the kind of materials properties that we might want. And so we can learn from nature about what we want to do, and then we can think about how to do that. Okay, so we have biologists finding new organisms. We have biochemists and chemical biologists understanding those organisms. We have chemists developing new ways of mimicking those systems in a synthetic, abiotic way, right? And so very much being inspired by the biology. But we can even think about taking that to the next level. And one of the things we want to do as part of the Integrative Sciences Initiative is think about how we can accelerate this process even more. And so enzymes take molecules, convert them into products. We just looked at that. That's a schematic, a cartoon of how that happens. And as part of our biomimicry efforts, we need to really harness the power of the enzymes themselves to do that. And so that's an exciting area in itself. But when we think about doing non-enzyme catalysis, so using those metal catalysts to do essentially the same thing, nature has benefited from huge evolution in those systems, right? And so for, millions and millions and millions of years, right? Billions of years, nature has evolved these enzymes to be really good at what they do. And so it's pretty outrageous to think as chemists, we're going to waltz into the laboratory and in 5 minutes, we're going to be able to mimic what nature spent, millions of years evolving, right? That doesn't really make much sense. So how can we increase our shots on goal to do that? Well, in 2023, it turns out that we're starting to have the capabilities of leveraging artificial intelligence and robotics to really make chemists a whole lot more effective. And so we can think about using robotics and AI to get an evolution type process, but now in the chemical lab. And so if you told somebody AI and robotics is going to be relevant to biomimicry, that might sound a bit disconnected, right? But it's not whenever you think about what you're literally using it for is to increase the speed of which you can mimic those biological systems. And so there's a lot of exciting opportunities here. And Malad over in chemical engineering is one of the leaders in this field. And so we have an exciting opportunity at NC State to continue to build on that, and we are working on that. But we can also mimic function, right? And so I'm talking about mimicking form. That's how nature does things. But what if we want to mimic what nature makes, right? And so these molecules like cyclosporine and the antibiotics and the vitamins we take, right? These are really important, right? But sometimes they're really complicated structures, right? Sometimes they are really hard to make. Sometimes they are really expensive to make, right? Or sometimes, even as a chemist, you can't make them, right? It can be a challenge. And so, I like to think about this topic, in terms of mimicking form and how we mimicked birds flying, right? We didn't make a bird because we all recognize the challenges in the weight of humans. And even if we had wings, we couldn't fly, right? We wouldn't have the strength to flap the wings that we would need in order to fly. And so when you think about mimicking, we built planes, but they very much mimic birds in many ways, except for the thrust, right? Instead of the wings flapping, it's the engines providing the thrust, but the rest of it is very much mimicked by how birds function. And so we want to mimic the outcome, but not necessarily the same form, right? And so this is the other angle of molecular biomimicry that I think is a huge opportunity. And so nature builds all of these molecules from a rather limited set of building blocks, right? And so if you go out and you dig up the soil outside, there's only going to be some limited amounts of molecules in that soil. That's what nature has to pull from in order to make things. And so you get some combinatorial assembly of whatever's there, right? And you can tweak it and change it and modify it, right, a little bit. But at the end of the day, natural molecules that nature produces are some combination of some kind of standard building blocks. But as chemists, we can essentially combine atoms in any way we want, as long as we can develop a way to do that, right? And so we're not limited by that anymore. And so if we see something nature makes that has an amazing effect, like attenuating the immune system or curing infectious disease, maybe we can make something that mimics it, but make it a lot simpler. And maybe we still use some of nature's systems. Maybe we still use some of its enzymes to do that. Maybe we still use chemistry we learned from nature to do it, but we're going to make something else. And so this is just, you don't need to understand anything about what's on this slide. All you need to do is connect colors, right? And so this is great for the chemically challenged. Because, you know, if you look across these molecules, this middle molecule here, right, is a molecule that has really interesting biology. It's called 4-ball. Right? This molecule has all kinds of interactions in a biological setting. You don't need to know much about chemistry to appreciate it's really complicated, right? There is a lot of atoms in this molecule. There's a lot of different arrangements of them. It's just really challenging. As a chemist, that's something I don't want to have to go make, right? But over a lot of evolved study and understanding of these molecules, thinking about the biological systems and the interplay between the molecules and the biology, it's been found that in order to have that biological effect, all you really need to do is position 3 atoms, oxygen or nitrogen, in a 3-dimensional space in a way that this molecule does it. And so these obviously simpler structures, I think everybody can acknowledge these are simpler without understanding anything about it. These are able to mimic that effect because they put the groups in 3-dimensional space the same way this does. Nature makes this molecule for a number of reasons, but one of them is because it's using a certain set of building blocks to do that. And yes, those building blocks are a bit on the complex side, but nature is able to handle that easily. Us human chemists aren't, and so we need to think about how to mimicking it and get effective functional products, but in a simpler way. And so there's, I think, huge opportunity to thinking about function and mimicking that and not just mimicking form. But form is also important sometimes. Sometimes you can't do this, right? Full disclosure, sometimes molecules of this kind of complexity require every bit of that complexity to have their effect, right? And so we don't have this luxury all the time, but sometimes we do. And sometimes it can be as simple as, okay, well, instead of making this, we're just going to make the one without this group on it, right? And that's going to be, simpler to make, and that can have the same function. And so that's what we can think about. And so, I think the future is really bright for molecular biomimicry. I think we can mimic both how nature makes molecules and what we do with them. Teams of scholars from the micro to the macro can contribute to this. I think, we need new sources of molecules. We need to find new organisms that are producing really interesting molecules, and we need to find out how those molecules are made and hopefully find new enzymes that synthetic biologists and biochemists can use to do really cool chemistry as well. And, I think a bit dramatic, but I think it couldn't be more true that, a single molecule can change the course of history. And, thinking about developing that, something as simple as, penicillin, when you think about the impact that antibiotics have had, not only on human longevity, but on everything from globalization to our food supply, I think is an amazing story from one little organic molecule. And so a lot of this is what we really hope to channel in this new initiative in building. I'm really excited to engage with many of you as we think about this. The building also has lots of interest in applying these molecules to infectious disease and cancer and the such. But I think at the end of the day is really fueled by this idea that we can leverage nature's ingenuity in making and using molecules to address a lot of our grand challenges. And so thanks so much.
Wondering whether, with your talking, Clint, whether the honeycombs on the ceilings need to be rounded. Amazing, Josh. I think we'll keep moving here. So our next talk is Gavin Williams. Where's Gavin? So Gavin's the department head of chemistry here at NC State, and Gavin's going to talk to us more about how we mimic the processes of nature to engender new molecules. Thanks so much, Gavin. And thanks again, Josh.
Thanks so much for the introduction, Rob. So what I want to do in my time with you this morning is to give you a pretty high-level overview of what synthetic biology is and what synthetic biology is doing. I'll try to argue the relationship between synthetic biology and biomimicry, and then I'll hopefully provide some examples which are explaining the relationship between synthetic biology and biomimicry, and could potentially catalyze some discussions later on to think about how, as a community, we could leverage synthetic biology and mimic nature in some new and exciting ways. This morning's talks are all focused on this idea of biomimicry, and I could summarize biomimicry as being some form of nature-inspired innovation for sustainable solutions. To contrast that slightly, synthetic biology is often defined as the design and construction and redesign of biology, and overall aims to make engineering of biology much easier. There's actually a little bit of a nice analogy between synthetic biology and synthetic chemistry, relevant to Josh's talk. Synthetic chemistry is such a established discipline. It's been around for well over 100 years, I believe, at this point, that synthetic chemists can design the synthesis of almost any complex molecule that they want from simple chemical building blocks. And the analogy is that synthetic biologists want to do the same thing with biology. So synthetic biologists want to be able to use the parts and pathways and components of biology as building blocks to create new things. And typically, the concept of biomimicry involves non-biological parts. I think, as we've seen with the honeycomb example, non-biological parts, devices and materials, and perhaps processes in that design process. Typically, synthetic biology, as that name suggests, uses things that originally come from nature, from biology. So these could be macromolecules or processes or mechanisms or ideas from biology to create new things. But what I'm arguing overall is that as synthetic biology continues to develop into a mature field, it's going to be able to create things that, although ultimately consist of things that might be familiar to biologists, proteins, DNA, RNA, that those things that synthetic biologists create are not going to look like anything that is originally come from nature. So that over time, I believe that the field of biomimicry will converge more closely with that of synthetic biology. And there's obviously a complex interplay between these two areas. But what both of these areas have in common is their desire to provide solutions to grand challenges of society related to health, the environment, and energy. So I'm going to try to give you a few examples to illustrate these points today. So nature is able to generate staggering complexity, and nature can also generate super molecular structures and function and complex phenotypes by leveraging the central dogma of molecular biology. So you're all familiar with the flow of information through DNA, RNA to proteins and enzymes, and those enzymes catalyze chemistry and make molecules, and those molecules do things inside those cells as well. And higher order structures and complexity emerges by design through these natural processes. What synthetic biology generally aims to do is to expand upon what nature has already achieved through many millions of years of evolution. So the scale of synthetic biology operates on a similar one found in nature, from the nanoscale through the macro scale. But synthetic biology can leverage natural genetic systems, perhaps genetic systems that use unnatural genetic building blocks in their systems to evolve new functions. Synthetic biology can also expand on the molecular building block repertoire that nature has arrived at. So there's no reason that synthetic biologists should be limited to the 20 canonical amino acids, for example. Another feature of synthetic biology is the desire to ultimately modularize and standardize all of those biological parts. So we want to be able to take promoters and operators and RNA devices and proteins and metabolic pathways and mix and match them in different ways, like Lego bricks, to make new things. Directed evolution is a key method used in synthetic biology, which I'll elaborate on in a second. And ultimately, in addition to building things that might have important applications, synthetic biology can also be used to explore and understand those underlying mechanisms and processes. So there's an adage that comes to mind, what I cannot build, I don't understand, and that applies very well to synthetic biology. I also believe that synthetic biology represents a true convergence of interdisciplinarity. So the most impressive examples of synthetic biology involve collaborations between people from genetics, chemistry, engineering, maths, biology, and even art and design, which I'll touch on later. And so I've listed on this figure the colleges which are likely to be involved in existing or future collaborations in this area. And I've also listed the departments which currently house faculty which are already collaborating in synthetic biology, but could be doing more of that in the future. So I think this serves to illustrate the potential breadth and interdisciplinarity of synthetic biology throughout the campus. Okay, so directed evolution is a really key method in synthetic biology, which borrows heavily from nature. It's a very good example of biomimicry. So directed evolution, as the name implies, is a process that mimics natural evolution, but speeds it up by many orders of magnitude so that we can evolve new molecules with desired phenotypes in the lab. The basic idea of directed evolution is that a large library of variants is created using molecular biology. And these variants are subjected to a high throughput screen or a selection in order to identify those relatively rare variant molecules which are displaying some property of interest. And then this process is repeated as many times as necessary to develop the goal of the directed evolution. There's lots of opportunities in this approach for people across NC State and beyond to have a really big impact in directed evolution. So we need new methods of diversification. We need to develop screening and selection technologies to find those rare variants. Directed evolution folks can collaborate with chemists to develop novel biocatalysts that can facilitate the synthesis of complex molecules. We can apply evolution to entire metabolic pathways to increase the complexity. We can involve computer-aided design in order to facilitate this process and make it more efficient. And we can also work with biologists and microbiologists and biochemists to develop approaches based on continuous evolution where the experimenter doesn't need to provide any input, but an artificial system does all the evolution in an autonomous fashion. A really nice example of the power of directed evolution was published a couple of years ago now, which was the result of a collaboration between scientists at Merck and a protein engineering company called Codexis. And this team were interested in developing an entirely enzyme-based route for the synthesis of an investigational anti-HIV drug. And so the team was inspired by a naturally occurring biochemical pathway called the nucleoside salvage pathway, which they felt would be able to produce the desired drug in a highly efficient process. The problem, though, that at the conception of this project, this was an entirely theoretical enzymatic route. And what was required was the directed evolution of 5 enzymes in this pathway in order to make this pathway work and make it a viable approach to making the drug. So that's exactly what they did. They subjected 5 of these key enzymes to many rounds of directed evolution, and the outcome was a highly efficient process that could take place in one reaction vessel to make this anti-HIV drug. So at the time this is published, this biocatalytic cascade halved the number of steps compared to the traditional synthetic chemistry approach to making this drug, but also made the desired compound in a much higher yield than the currently published synthetic chemistry approach. So I think this is a really nice example illustrating inspiration from nature in designing an enzymatic route to a drug, but also inspiration from the natural process of evolution to actually make this process a reality. Stepping up the complexity somewhat, synthetic biologists have more recently developed artificial photosynthesis by leveraging synthetic biology in a different way. So in this example, the synthetic biologist designed and then built this 16 enzyme sequence of chemical reactions. So these enzymes are found in nature, but they're involved in metabolic pathways that have got nothing to do with photosynthesis. So the authors of this study selected these enzymes from nature and stitched them all together and showed that they could fix CO2 and make some ATP and provide some key metabolites, as does photosynthesis. And since that initial discovery, the synthetic biologists teamed up with some engineers to take this a step further. So they're able to, for example, purify the biochemical material from plant thylakoids, and then take the 16 enzyme designed pathway and pop that into that thylakoid extraction, and then house all of that in vitro material in a microfluidic device. So they made little emulsion droplets. So they're able to then demonstrate that those droplets could use the light energy to then catalyze this artificial form of photosynthesis in these artificial cells. So this is really pioneering work in my view, and it has broad future applications related to energy storage and capture and perhaps conversion as well. In another example, synthetic biologists have for some time now been engineering artificial bacterial strains that can be used to detect and in some cases treat cancer inside living organisms. In this pretty simple early example, an engineered E. coli strain was fed into a mouse that harbored liver tumors. And the synthetic biologists demonstrated that these E. coli, these engineered E. coli strains as a probiotic, could actually target the tumor cells and inhabit the tumors. And these cells were also engineered to produce fluorescent reporters. So they're able to detect the presence of very, very small tumors in these animals by measuring the fluorescence in the urine of these animals. Since then, synthetic biologists have taken several additional steps by building complex genetic circuits so that these engineered bacteria have got greater specificity in the tumor detection and can even detect the tumors in situ without having to detect them in the urine, for example. And there are many other examples of interfacing engineered bacteria in this way with other detection methods and even abiological devices for cancer therapeutics. In other more recent work, synthetic biologists are leveraging their ability to engineer complex signal transduction pathways to actually manipulate the developmental processes and underlying morphology of organoids and embryos, for example. So typically these efforts rely on finding in nature proteins that respond to different frequencies of light and change their shape, or ability to interact with other proteins. And so those light activating domains are transplanted from their natural source and used to create these light-triggered transduction pathways in organoids. So these have recently been used to alter the morphology of organoids in different ways. So this provides the ability to manipulate the structure and function of artificial organs, but also to delve into some of the underlying biological mechanisms. With, I should say, the ability to control those processes with exquisite spatial and temporal control. So I'll end with a few aspirational examples of what synthetic biology aims to do. One example is to create so-called living cities. For example, you could imagine trees to be genetically engineered by synthetic biology to produce fluorescent proteins. So we could replace street lamps with fluorescent trees. It's a little far-fetched, but I guarantee that people are actually trying to do this right now. So that's very exciting. Extending this approach, synthetic biologists are working with engineers to think about how we could literally grow a home from a brick. So you could imagine some kind of a brick built from fungi that contains all of the genetic information to program the formation of a home. It's not going to be a home that looks like homes that we live in right now, but these folks are interested in how we might terraform planets in the solar system, for example. And then synthetic biologists are also interested in augmenting or completely replacing civil engineering materials that are used today. So there are already companies that are making bricks out of fungi material and using those to create structures. Synthetic biologists have also spun out a large number of companies that are developing various interesting technologies. One company called Bolt Threads has recently developed engineered yeast strains that make an artificial form of spider silk. And this company actually teamed up with fashion designers to incorporate this artificial biomimetic material into actual pretty nice looking clothes that you can actually buy from their website. This company is also using fungi to make an artificial form, a sustainable form of leather. And then artists are also taking a lot of inspiration from synthetic biologists. And in many cases, synthetic biologists are working with artists and designers directly to make their ideas a reality. So the image on the right is an artistic rendering of the vision of the future of plant synthetic biology. So this is a aspirational artificial plant that is engineered to overproduce some vitamins in the leaves of this plant. But this artist also wanted to visualize the concept of using the root system of the plant to make novel textile-like materials. So again, a little aspirational, a little far-fetched, but a lot of this is very quickly becoming a reality. So in closing, I think there's a lot of potential for synthetic biology to impact almost every aspect of our day-to-day life in the future. It's clearly heavily inspired from what already exists in nature. And I'd just like to encourage everybody to really explore your imagination and just think about how you might be able to apply and leverage synthetic biology in the future. So thank you for your attention.
Thank you so much, Gavin. I think we have time for one question, and then we'll have David Hu come up and talk.
Hey, so I have both a question and a comment with the last two talks, which are absolutely spectacular. Which is nature makes a lot of things that help us. It also makes a lot of things that kill us, as do synthetic chemists. So there's like a real opportunity, I think, in here with this interdisciplinary approach to bring in the idea of sustainable chemistry and reduced toxicology. And so I sort of see that as an advantage, another advantage of bringing biologists into that space, as well as the ecologists, because if you make glowing trees, maybe the species that live on the trees won't be totally happy with that. I don't know. But I'm just wondering how toxicology and sustainable chemistry fits into these imaginative designs that you've presented.
Yeah, thanks,
question.
Heather.
Thanks,
That's a great point. I do think that in terms of synthetic biology, one of its long-term goals is to be a sustainable technology for the future. So elements of sustainability are generally considered in those processes. And I think that applies to the field of synthetic chemistry as well. There are I think I saw Lucia walk in earlier. Oh, there he is. So we have chemists at NC State who are very interested in making synthetic chemistry more sustainable. And to address your earlier observation, I think I forgot to put this on the slide, but the genetics and engineering center at NC State is very involved in facilitating discussions around the ethics and societal impacts of genetic engineering and synthetic biology and related sciences. So there's a really important part for those folks in these discussions as well. Mm-hmm.
Thank you, Heather. Thank you, Gavin. One more round of applause for Gavin. So our next speaker is David Hu. Well, I just saw, where'd you go? There you are, David. And so David is really, I think, one of the most creative researchers doing biomimicry work in the country. He has an amazing book, and I just had to look up the title again so I remember it, How to Walk on Water and Climb Up Walls, that I highly recommend. And Dave's going to talk to us today about some of his biomimicry work and some of the many dimensions he's exploring. Thank you so much, David.
Thanks, Rob. I thought we'd start the talk with a stretch break. Everyone stand up if they want to for like 30 seconds while I introduce myself. So I'm actually in mechanical engineering and biology at Georgia Tech. And I started my career in the math department looking at how insects walk on water. So what you're seeing here is what I tell my kids are two parents having a discussion. And this is completely happening on the water surface. These insects are actually denser than water. If you add a single drop of soap, they would all sink to the bottom. So this is made possible by the fact that each of their legs is the width of a human hair. And each leg is covered by 10,000 hairs per square millimeter. It's actually the hairiest surface in the world. And what that does is it creates a really, really rough surface. And it takes advantage of of the forces of surface tension. Surface tension is what keeps bubbles around, and it will allow these insects to basically walk, jump, climb on water, all without breaking through. So that's all like it if it were like land. And this is me back in grad school doing this high-speed film. This is a view of what happens underneath the water surface. So these insects, to engineers, they posed a huge number of questions. First of all, they're called water striders because if you watch them there's 90 species of ants around this campus, and there's definitely water striders in your lakes and ponds. But they're called water striders because of their ability to glide. They're almost frictionless, and that's because their legs are so hairy that only 1% of the leg actually touches the surface of water. 99% of that leg is actually covered in air. And so they're basically gliding on a cushion of air, just like an air hockey puck. So the question arises, how do they actually push themselves forward? How do they actually move? Especially if you look at this leg, it's pretty much the worst oar you can imagine. Imagine going to a lake and someone giving you two big pieces of spaghetti and asking yourself to row across the lake. How are you going to get across? Well, their secret is that they use their legs as oars, and every time they push down on the water surface, the legs are so water repellent, they generate these dimples, these sort of air-filled cavities. They're called menisci. And they use these menisci as the blades of the oar and their legs as the oars. So every time they row, they generate their own blades using these air pockets. And that's what generates these patterns in the wake of these insects. So that's something engineers would never have figured out. And the engineers have loved this so much, they actually put this on the cover of the fluid mechanics textbook that I teach with when I came to Georgia Tech. So this is actually what freshman, sophomore engineers start studying for their curriculum. So, and there's been many speakers that are talking about diversity. And there's 1,200 species of these insects all over the world. going from, you know, as small as a couple millimeters to the giant Vietnamese water strider, which is as big as my hand. That water strider is really pushing the limits so much of how much weight it can support that it actually has a safety factor of 2, just enough for mating. And then after that, it'll sink through the surface. So we basically took so we've been taking the designs of the legs. You can see they have very long legs for rowing and for weight supports. As chemists have been taking a lot of inspiration from the leg itself. So for example, this is the leg that I was telling you about. The 10,000 hairs per square millimeter, you can only see them with a microscope. And features that you can only see with a scanning electron microscope. This is a single hair. These hairs are covered with grooves that act like a little water strider leg in itself. And this is a common principle in nature to make hierarchical structures. We still can't really do it in engineering, but we're trying to, where we basically cover each one of these hairs with tiny little grooves to further increase the storage of air so it can be water repellent. So based on what we saw, we built this device called RoboStrider. It's made of lightweight stainless steel and aluminum, and it supports itself just like the real water strider. And it's different from a robot because it actually doesn't break the surface. It stays on the water surface like the skin of a pudding, and it rows just by bending it. So you can imagine it doesn't get wet itself, and it can, if we had designed it with more water repellency, it would glide for long distances. So this was the first sort of water strider inspired robot, but it really started this long journey by engineers to build robots that can walk on water. And it's not that we're going to have robots replacing boats. It's sort of just an engineering challenge that is basically inspired building really lightweight materials, low weight batteries, and really just pushing the envelope of what we can do with small things. So for example, here, this is in the last 15 years, all the different water strider robots, the ecology of robots that have been evolved to sort of sit in the water surface and try to repeat what you saw in that first video. And these are just some of the innovations on trying to build legs that are like these water striders. Instead of mimicking the individual chitin, the proteins, they're actually using sort of carbon nanotubes and making a forest that is really, really water repellent. In work that we've done more recently, we've looked at how insects can actually build some of these water repellent surfaces without having hairs themselves. And this is a creature maybe you're familiar with. It's invasive. It's called fire ant, known because when they sting you, it feels like you've been burned. They actually are not hydrophobic themselves, but when they link themselves together, they can basically build a structure that's kind of like the water strider leg, very similar to Gore-Tex, where air is trapped between each individual ant, and these ants together are water repellent. Individually, again, they're not actually very good swimmers, and they would sink, but you can see how difficult it is to actually push this water strider, this ant raft underneath the surface of water. It's helped by both the air trap between the ants, and also the fact that this raft is very, very flexible. And this is actually the grand challenge of robotics, to build this veritable bucket of parts that you would dump out in the ground, and they would crawl around each other, and they would link together to build a more capable device. For example, at UPenn, this is a sort of a ant raft inspired device that has small linkages, just like the ants, to connect to each other. It has some flexibility and allows sort of larger objects to cross. This is the ants building a similar structure. They can actually sense the deformation of the water surface as it gets to land, and they can start doing a tank tread as they sort of roll towards land. So this is one of the 4 stories I want to talk about. You'll know I get to the end of my talk because I'll be talking about wombat poop. And I actually has anyone heard of wombats and their poop? Do you know they're cubic? Yeah, they're cubic. So we'll talk about that. And here, I'm going to start passing it around. By the time it gets to the end, I think we'll be at the end of the talk. And feel free to take selfies with the wombat poop. So diversity is really important for looking at larger animals. And we haven't actually talked too much today about vertebrates. And you know, probably most of the vertebrate knowledge you have is from dogs and cats. But cats are actually from a family of 13 different species all around the world. And one thing they do very well and very much in common is they sleep a lot. And the other thing they do is they groom themselves constantly. They are probably one of the cleanest animals out there. But up until a few years ago, no one really knew how cats groom. This is actually a high-speed video of a cat licking a carpet soaked with cat food. And you can see its grooming structures are completely in its tongue. So the tongue is actually like it's like a comb. It's a washing and drying machine. It releases enzymes. Saliva is actually one of the few substances we can't mimic because it has over 20 or 30 different individual compounds that dissolve blood and oil. And allow this cat, so we've actually measured this, a cat uses 3 tablespoons of saliva every day to clean itself. When we take a bath, we take about 10, 20 liters of water, but the cat can basically do it with just 3 tablespoons. And that's made possible by the fact that the cat's tongue is covered in individual spines. That's why if you've been licked by a cat, it feels very different from if you've been licked by a person. Very, very rough. These are, so the Tennessee Tiger Haven is a place where tigers, when they're illegally purchased in the US, they go to retire. So we actually went to their freezer and raided it, and these are all the cat tongues we were able to get from their freezer. They're now sitting in my office to scare students when they come by begging for grades. For example, this lion tongue is as tall as your head. It's huge, and tiger, snow leopard, cougar, bobcats. But one thing we've been talking about today is convergent evolution. We've seen a lot of talks where a lot of these animals and insects and plants have come up with ways to derive the same thing sort of through independent pathways. And one thing all these animals have in common, these cats, is that they are covered in the spines that you saw. And up until a few years ago, no one had visualized, I mean, this is a spine that's sitting in your house cat's tongue, and there's millions of house cats all across the country, in almost every house. But people did not know what was on their tongue that allowed themselves to stay so clean. This is the structure. It's made out of keratin, similar to your fingernails. And it's kind of got, imagine a round base to sort of stay embedded because these lions are licking their wounds and licking bones. But it also has a skateboard ramp, a very sort of curved surface at the top. So as a fluid dynamicist, I recognize this surface as one that automatically wicks up water. So this is what happens every time the cat puts its tongue in its mouth. Every time it sticks it in, these spines act like a sponge, where if they're made out of the appropriate material, the saliva, which acts as a cleaning agent, is automatically wicked into each of these pipes. And these pipes are so tightly wound around the saliva. They're small. They're only a fraction of a millimeter thick, that they can't get dislodged easily unless they hit the right key. And for cats, the key that goes with their lock is their own hair. Their hair actually fits exactly through these spines, so every time they groom themselves, they don't release any water on the ground. Every time you take a shower, you're mostly throwing the water on the ground, but here the cats are releasing the saliva precisely on the surface of their hairs. And that is exactly what's cleaned, and that's why they can clean their entire bodies for only 3 tablespoons of water per day. And I'll remind you, a cat, their hairs provide this tremendous surface area that could not be cleaned otherwise. For example, a kitty cat, if you add up all the surface of its hairs, it adds up to an entire ping pong table. A beaver, with its millions of hairs, has a surface area of an Olympic ice skating rink. Imagine trying to clean an entire ice skating rink with just a couple tablespoons of fluid. But that's what these cats do. The cats, when we looked at these cats, it also inspired us to look at how hairbrushes are designed. And I mentioned this in the title. And for years, we've really considered hairbrushes as sort of flat, sort of rigid objects. But nature has come up with all sorts of sort of soft ways to build things that is just being looked at. For example, these soft materials that are underneath these cat tongues, we built in the silicon mold, and we embedded these 3D printed cat spines. And we found some interesting properties. For example, if you take a rigid hairbrush, for example, maybe if you're bored on a Friday night in Raleigh and you want to clean your hairbrush, you'll find it's incredibly time consuming because these bristles are rigid and the hairs have to be sort of removed just individually one by one. In evolutionary terms, if cats had to do this, they would have died a long time ago if their tongues got basically stuck with hairs all the time. So instead, because the spines are all facing a single direction, what the cats can do at 1 a.m. or 3 a.m., anytime they want to make your life miserable, they just hawk up this hairball by rubbing their tongue on the roof of their mouth, and they can throw this up into your slippers at any time of day. It's just basically anytime they want to do that, they can create their hairball. So when we published this, there's a lot of interest in basically these self-wicking modules on the cat brush. For example, this recent paper on making these nasal swabs that we've been using for COVID testing was inspired by these cat tongues, where each of these nasal bristles would wick up more saliva than just regular having individual bristles. And we've been also talking about L'Oreal. They're really interested in products that use less water. And Nike, who's interested in having swim caps that sort of don't fall off your head. And there's some doctors interested in how cats remove debris from their wounds. Currently in surgery, they basically just take a scalpel and just remove the entire thing. Cats actually, they massage the materials out by using the hard spines. I have about 5 minutes left, right? Okay. So I want to talk about a little bit about my Ig Nobel Prizes. The first is about animal urination. So this is the Atlanta Zoo. This is how we study urination. So the big question of urination is, how do so many animals get it out when there's such different sizes? For example, you probably haven't seen this before. This is what a rat looks like when it's urinating in your backyard. It actually cannot even generate its own urine stream. And that's because the surface tension of the fluid, the same that supports the water shredder, actually pushes it back in the body. And that's why they can't even get a coherent jet. So that's the best they can do. In about 40 minutes, when we go for a coffee break, you'll probably generate a structure like this. This is a goat's urine stream, where again, surface tension acts to create these small drops. If you're a milk farmer in North Carolina, this is probably the last thing you want to see coming at your face. This is sort of what we call a fluid fish bones of urine. And this is my favorite video of all time because it's part 1 and part 2 of my student's PhD thesis, all in the same video. I was super happy because we wrote 2 papers based on that one video. So for engineers, what inspires us is how all those animals can eject fluid in such short amounts of time. For example, the elephant, which was the most impressive animal there, it actually has 20 liters of fluid in its bladder. That's an entire garbage can. And it gets it out in the same amount of time as these other animals, and that's because it releases it at the rate of about 5 shower heads going on at once. So the way that happens is that these animals have taken advantage of what we call beer keg physics, where they have a structure in their body called the urethra, which I call the pee-pee pipe, that basically allows them to arrange their urine vertically and use gravity to push it out. So female elephants, for example, have a 1.5 meter long pee-pee pipe that allows fluid to come out very quickly. If you don't believe me, you can take containers and mimic the pee-pee pipes of different animals, and you can get arbitrary large sizes to be removed in short periods of time. So this looks like it was just for fun, but actually has resulted in these new kind of medical procedures where, for example, now in Japan, to figure out the bladder pressure, they'll actually just ask you your pee-pee time. This is a Japanese doctor who interviewed 2,000 Japanese people to show that as you get older, your bladder decreases in pressure, and you can record that change with the pee time. If you ever have urethral damage, you should be comforted that urethras that are made out of collagen in human skin are now actually designed based on this 21-second rule for urination. Animals urinate for about 21 seconds, and they make sure that these can last 8 21-second durations per day. So the cubic scat, which is going around, is actually quite cubic. They don't have wombats in Atlanta. We have to go all the way to Tasmania. So this is our collaborator getting his hands dirty and catching these wombats. What you imagine, they're actually like squat, really muscular toddlers. They're the world's almost cutest animal, second most cutest animal. Number one, of course, is the panda. And they live their entire lives in underground burrows because of the drought-ridden conditions. Their physiology has changed so much, so they're marsupials, but they actually point their pouches backwards. And the unfortunate consequence is that every day they poop on their babies' faces about 100 times per day. So why are these scats cubic? One of the biological reasons is that to warn their neighbors and to mark their territories, they basically all poop in particular places called latrines. And we think that if they all pooped on top of a short stump in a rock, if it were circular, it would fall down. But if it's cubic, it stays on top of the rock. Now, how does it stay cubic? Well, the number one reason, if you ask Australians, is they have square anuses. So making poop should be like making spaghetti. But actually, if you look at their anuses, they're not particularly round. Instead, the answer is much more sinister. This is a poor wombat that was hit by a car, which happens almost every day in Tasmania, which we procured. And in its body is a cube-making factory. And engineers see this as fascinating because they take a round hole and make a square peg. How in the world are you going to make corners out of something that's basically smooth like this? And the way they do it is two reasons. Those hexagons you saw from Clint's talk, they actually reappear in structures in nature. For example, this is a giant causeway in Ireland. These are cornstarch, drying cornstarch cakes. And the basic idea is that these kinds of materials are solid, but as they lose water, they lose volume. And if they do so really, really slowly, they can have constant length. And that's what causes these things here. And that's what caused the wombat's poop to have basically constant length. But what about the four corners? So to study that, we hung the wombat intestines like a gruesome Christmas ornament in my lab from the ceiling. And we saw that we actually measured them. We found that they're kind of like Rolo candy. And if you ever eat Rolo, you'll never see it the same way again. In that everyone thought intestines were a constant strength throughout. But actually, wombat intestines have a band that's 4 times stiffer in basically two of these edges. So by basically simulating this, we can show that, imagine a band is like a stiff calamari, an overcooked calamari. That will contract first. The remaining regions get sort of left behind because they contract slowest. And by having these bands, they can basically create these corners. So we think one day if you go to the doctor and you have abnormally shaped feces, they'll be able to diagnose that a little bit as having stiff colons, and that's actually associated with colon cancer and other things. But if you definitely have any kind of cubic feces coming out, you should probably talk to your doctor. Okay, with that, I think I gotta wrap up. Thanks a lot.
Thank you so much, David. I think we'll transition right to the next speaker. Dr. Renee Lopez is from the Department of Physics and Astronomy at UNC, is going to talk to us about butterflies and photovoltaics. And so we're covering vast terrain of biomimicry, from the molecular to the butterfly to the square poop. And
you cannot lose that.
I don't know what to do with that transition. Thank you, David.
Well, it's gonna be hard to come back from that. Follow this act. All right. Let me say that I'm very happy to be here. It's not a far trip from home, but I don't make it very often. Thank you for the invitation. So I'm gonna tell you about my research, or what I have been doing in the last few years, and how it connects to the topic today. So this is me. Happened to land in the catalog of the UNC a few years ago. And I have a research in different topics for over the years. I work in materials, particularly oxides, for some interesting physics applications. I also work in solar fuels. I've been doing nanostructures for some chemical sensing. And the last things I was doing, maybe up to the COVID, I was working in solar cell technologies. And also I had a research topic in butterflies. As you see from this list of here, the only one that seems very odd, doesn't seem not to fit, is actually a butterfly. So how did I get in that racket, in that business? This butterfly in particular is one that is famous out there. The morphoretenor is very blue. If you have gone to the butterfly houses, I think there was in Raleigh and Durham, they frequently have them because they are really striking, beautiful, and impressive. In fact, the projector doesn't make justice to the butterfly. I mean, here it looks blue and looks pretty blue, but when you look at them in real life, it is strikingly unique in coloration. Nothing man-made can actually look like that. And the reason for that coloration is because the structure of the wings has these nanoscopic features. And it's very unique, okay? Although other butterflies are not as strikingly beautiful, the pattern, this kind of Christmas tree, is actually common in many species of butterflies. In fact, a vast majority have this or another sort similar structure. Some butterflies combine pigments, but this one is mostly completely due to the nanostructure that has the color, what make it very, very interesting to study, I would say. So the reason why this is supposed to function as a coloring device is a study by biologists and people that do optical design for many years. And the conventional explanation is that there's a interference effect between the different branches of this pine tree structure. Sort what you see here in this diagram, okay? And I was, as many people, happy to take the explanation, and I thought it was nice enough. But I was interested to see whether we could actually characterize whether this blue coloration, right? You look at with the eye, it looks pretty and nice, but when quantify it. So we built this small apparatus here that you can see in the screen. And what it has really is a source of light, and then it has a collector that is actually at a variable angle. And right in the center, we mount the butterfly, and you can actually study the reflection of this at every different incidence angle and collect also light at every possible reflective angle. And what you see here is this pattern, right? So you see in the bottom is the incident angle of light, and in the vertical axis you have the scattered angle. And what you see here is actually a very striking pattern. Cross diagonal, marked with an M, is what you will see if you put a mirror right there in this machine to measure, right? So you can see the butterfly reflects light not at all like we'll do a flat surface or a smooth surface. It's actually totally the opposite. The light is actually back reflected completely. And that's a very unique feature. Incidentally, you can see that the butterfly also appears in the pattern, which is it was a nice thing to see. But the main point is that this is what makes the butterfly color very special. It's not only that it's blue, but it's actually blue and has a very particular reflective quality that is not common to any other, say, common surface that you look at. So we decided, okay, can we actually replicate this, all right? So I and some students were able to squeeze some money out of projects and try to make an effort to replicate the structure of the butterfly by artificial nanofabrication. So here it just goes through many, many steps, but the main goal here is at the bottom of these two sequence, you have a process to create a masters, and then we were trying to replicate this in a polymer. The idea here is that although with the master we can actually make the pine tree structure, we are bound to use two different materials that will have kind of different optical properties. The butterfly makes this with only one material. So the whole goal was to actually replicate in totality the effect with the butterfly, to create this structure that you see here at the bottom of the structure down here. And we went for it. So here are some micrographs of the initial steps. So then there is our attempt to make a pine tree. The butterfly, this is not quite not angular, but has the right staggering sequence. And then we went for the replication. And these down here, as you can see, are actually in one polymer. So this is not two materials, this is one material, and you can see the rows mimicking what the butterfly structure wing is supposed to be. And I said, okay, we got it. Seems we can actually go and claim success. And we measured the reflectance. It was well designed. This is a spectrum similar to what the butterfly does. But then when we put it in the angular characterization, we found it was not at all like the butterfly. In fact, as you can see here, the highest intensity is exactly what you will reflect with a mirror surface, with a flat surface. So just to remind you, let me point here this one. So you can see, although it looks blue, apparently, in fact, they look pretty similar at certain angles, is not at all what the butterfly does. It doesn't have the same optical properties. And that's when we say, okay, what is really making this thing work? Because it's not just the pine tree structure. There has to be something else behind that. Here is again the structures that we have far away, and put a student to actually do this simulation. You can see in the left is the experiments, and in the right is the simulations. And indeed, well, we got what the simulations predicted. And then we tried to simulate truly the structure of the butterfly wing. And then you see what the butterfly actually does. So you see this structure here has similar features, but it's not quite the same. As you can see, there's some staggering. They are not really in the same level, these branches of the tree. So I thought, well, is this an important feature or not? So we remove it, and actually it was still there, okay? To make the story short, after a lot of going back and forth, what was really the reason for this behavior? Turns out that the butterfly, besides having the pine trees, has another feature. The pine trees are incoherently related to each other. They are not truly periodic. And what we were building initially was truly periodic lines where all the trees were working together, creating very strong diffractive effects. Here in this other experiment, we had the pine trees. These ones really look kind of conical. And we made an effort to specifically make each distance between the pine trees be randomly connected to the next one. So they were really incoherent. In that case, each tree was functioning as one independent reflective structure. And then we got the pattern where we're looking for, where you have these lobes, and then the back scatter reflected and not in the mirror reflected, right? So all came really to matter, but the staggering of the branches of the tree, it was not an important feature. It was actually non-periodicity of the structure. So we were this level, and then decided, okay, so this is very nice. What can we do with this that is actually more useful than just the understanding of it? And we were into solar cell business. And any solar cell has to function with the two key parameters. One of them, I'm going to highlight it here, is the absorption of the light. That is characterized also by a distance, how deep the light can penetrate. And then there's a distance that characterizes how good are the charges to get out of the solar cell. If these two guys are very mismatched, or you don't absorb enough light, or you cannot extract the charges out of the cell. So with those designs in mind, you have to keep this more or less good. Otherwise, you are not going to get a good performance device. All right. So given the properties may be set for a set of materials, what we want to do is actually control the geometry. Kind of what the butterfly is actually doing, controlling the geometry. So we went to this toy design for a solar cell, where you have a flat cell that has your certain thickness, and then what will happen is this flat cell will have a structure inside that mimics the butterfly structure. And we saw that the absorption is supposed to be this level for a flat slab, but if you start to increase the pine tree structure inside, you can see the absorption starts to increase and has to have some features that peek out of there. And that's what we're trying to exploit of here. We went to fabricate a real device out of this idea. We omitted here the pine tree structure for this other model because for sake of fabrication. And we apply all realistic materials of there. You see, it's way more complicated than the toy model. And what we predicted is that the solar cell flat will have this performance, light absorption versus wavelength. And this solid blue line corresponds what you will get if you actually make it with this pattern structure inside. This is for 1D periodical structure, and this is for a 2D periodical structure. In both cases, you see this supposed enhancement in the light absorption. And there could be some features here very indicative that you actually are trapping more light in some what we call guided modes inside the structure. So that was the model, and get some funding to try to build it. This is the results we got from that. These are actually measurements of the reflectance of the solar cell. And in these other graphs down here, you can actually have some quantification of the performance of the solar cell versus different angle of incidence. And we did see some enhanced features here at the edge. You can see it's more rounded and a steep performance. And a little bit of the features that we have here representing the quasi-guided modes, kind of like here was predicted in the model. All right, so this is where all the light gets absorbed, some wasted in different materials, some actually wasted in reflection, and here, right here will be the trapped mode inside the structure and appears up here. So it went very well, I would say, as a first attempt. We were using, this is now our old-fashioned combination of polymers. And then we tried to recruit the help of mathematicians to tell us, okay, seems that putting some structure inside the solar cell will help. What should that be that structure? So here are the equations that are supposed to govern all the electronics and optics of this device. And there you have our friends from the mathematics department play with the structures inside the solar cell, kind of representing the butterfly wing structure, or going pretty crazy and whatever they could mathematically introduce, and produce this set of results. Okay, so they say that each of these dots represents a different structure, and you can see here is different levels of ratio of absorbed power to output power. This kind of representing the electrical efficiency of the solar cell, and here the efficiency, the optical absorption, and then here these lines represent overall power conversion efficiencies. And you can see there's tons of structures that actually are bad or not very good in reality, but up here there are a handful that actually stick out because they are more efficient. All right, these are not big different performance numbers. As you can see, the bulk of the pack is about a value of 0.03, and the best ones will be here at a value of 0.05. So small numbers, but actually shows that the structure really matters here. And we say, well, can we actually try to make one of these guys? So we went for it, but this time we picked a different material that was more possibly a better performance than the original one we tried the first attempt. And let me show you that one. And this one, the design, we're going to start comparing with a flat cell, but this is a solar cell that has actually quantum dots as the active media. Again, there are many materials up here, but the actual that actually does the light absorption is this lead sulfide. The model for this structure, you see just a flat solar cell with all these other components that need to be there for the electron capture and management are represented in this space. And this color code of here is actually where the light is absorbed when you shine the light from the top. Well, actually looks from the bottom. And this curve is the voltage versus current. And you want that this area under this curve be the largest as possible. Because up here, somewhere here where get the maximum voltage and the maximum current, that will be your power peak performance and power extraction. So this is a flat solar cell. And we play now with a one that is a little thicker. You can see that you almost get no improvement anymore. And that's because when it gets a little thicker, the charges really have a problem to get out. You may absorb more light, but you cannot extract them out. There's a limitation into the performance of the material in transport charges. Then here comes the structure, right? You can see you just get a structure, this kind of, how to say, pattern with a little grating up here. You get a slight improvement, which is not a lot, but you keep increasing that, you get more, and then you can get more. All right. So you can bring a solar cell to almost double the photocurrent with the same material characteristics. Okay. And you can see here where the light is absorbed the most is actually in the back of the cell. But without having all these things to do the optical light management, you are not able to do all the benefit of the structure. So all this is just for the optics, maintaining a limited thickness here to extract the charges. Okay, so that's basically what we were able to model, and then we went also for the fabrication of this structure. Let me show you the micrograph. Fabrication was not easy. Now you need to coat all these conformal layers in a non-flat structure. So the usual method to deposit these materials, this spin coating, was not amenable, didn't work. So it was pretty bad. And then clever student realized that actually we can try to do deep coating, where you actually go for the position where you put your substrate inside a bath, and then you extract it up, and then the solvent is evaporating and it's supposed to be coating nicely along the extraction lines. And that cannot work. You can see up here, these are microstructures. This part up here at the bottom is the conductive thin transparent oxide. But the first layer that is a little darker gray is already the layer of quantum dots coating very nicely around the structure. All right. So we thought, well, great, it's doable. So we went more for it. And this is a finished solar cell that incidentally has this kind of mouse traps up here, mouse holes. But this one is the bottom layer. Here you can see the active layer, the quantum dot layer. And up here, there are the electron collecting. And finally, up here is the metal on the top. And then we try to measure the performance. You have here the comparison, right? So this is, again, the external quantum efficiency, how many electrons you really get out of this thing per photon that you absorb. You have here the different colors of the photons. This is actually, as you notice, is a very good material for solar cells. It really absorbs from the blue to the near infrared. And the blue actually is so strong absorbing that there's no really need to help it much. You cannot really improve over what is already very good. But you can see here where the absorption of the infrared is weak. This pattern structure has a considerably better performance than the flat one that just peaks here about 20%. This one actually was more than 50%. And here are the IV curves, the current versus voltage for these solar cells. Okay, so this has been my journey working with these structures. I actually remember that I got inspired for coming to a seminar like this. In fact, I think maybe it was here in NC State many, many years ago. The first time I heard about these butterflies and the interesting color they have and how I get into try to fabricate them and then going to try to use this inspiration to work something for a solar cell technology. So the path has been like this. Get this idea, try to make the artificial reproduction, and then inspire this to bring it to the solar cell, try to make the solar cells, and see that in effect, you use this design idea, you can actually bring improvements to this material. With that, I just want to stop here, and I want to acknowledge the people that work on these projects. This is not my group today, but this is actually the guys that one way or another work on these projects. Thank you so much. Any questions?
I think we need to move on, Renee, but we'll have maybe some time for people to connect during the break. Thank you so much. So our next talk this morning is Dr. Ashley Brown from the Biomedical Engineering Department, which is joint between NC State and UNC. and she'll be talking about synthetic platelets for wound healing. Thank you so much, Ashley.
Great, thanks. And thank you, Rob, for putting this together. It's been really exciting to hear all the talks this morning and think even more broadly about all the ways that biomimicry can be used and just the vast array of applications. So I've been really excited to hear that. So today I'm going to give a discussion of a project that's been going on in my lab for a number of years now, where we've been using biomimicry to address key medical problems, particularly with regards to hemostasis and wound healing. And so broadly, my lab is interested in developing biomaterials for tissue engineering applications, again, mostly focused on wound healing. And really, tissue engineering as a whole is all about biomimicry. So how can we create constructs or materials outside of the body that replicate biological function to replace damaged tissues? And so this is, from that point of view, is really where my lab is coming from. And so I don't have to, really belabor this morning because we've heard this in so many talks. But of course, biomimicry is really just an approach that seeks to address human challenges by basically just emulating nature. And so the approach that we take is mimicking cellular function within the body. And so I'll get to that in a minute. And again, the primary challenges, human challenges that my group is trying to address is problems related to wound repair, bleeding, and chronic wounds. And so this is a really wordy slide, but basically wound repair is an essential process that we all have experienced at some point. That's important for tissue homeostasis, but it can go awry in numerous ways and lead to numerous different pathologies. One of those ways is if wound repair is uncontrolled, you can have uncontrolled bleeding, particularly after trauma or surgery. And this continues to be a major clinical problem. And in fact, uncontrolled bleeding after trauma is the #1 cause of death for people under the age of 40. And additionally, chronic wounds, so this is even after bleeding is stopped, chronic wounds can develop where the tissue doesn't repair properly, and chronic wounds in and of themselves are major problems in the US and globally. And so again, my lab is really focused on mimicking nature in order to address bleeding as well as chronic wounds. And the primary inspiration that we use are platelets. So platelets are blood cells that we have in our body, and many animals also have platelets. Platelets are the primary cell in your body that are responsible for clotting. So they're one of the first cells on the scene immediately after you have an injury. And so just a little bit of background here. This is a picture of false colored, but this is an actual picture of platelets shown in this purple color interspersed within a blood clot. Other key components that you see here are, of course, the familiar red blood cells, which are false colored red. And then you see them interspersed in this fibrin fibrillar network. And fibrin is going to be really important in this talk. But fibrin is the primary protein component that makes up the blood clot to stop bleeding. And so platelet-fibrin interactions are extraordinarily important in the initial clot formation to stop bleeding. But as you proceed after cessation of bleeding, fibrin-platelet interactions are also really important for sustaining and supporting longer-term wound healing responses. So in particular, fibrin-platelet interactions form that initial plug. Platelets also strengthen and mechanically stiffen the fibrin network over time. So that makes the network to support subsequent healing more stable. These interactions also facilitate inflammatory responses. And over time, these platelets also retract the clot. So they pull on the clot, which again increases stability, but also promotes healing. So immediately, a little bit of background, a little bit more background about platelets. So in the absence of injury, platelets are these small anuclear cells. They have a round morphology. They're about a micron in diameter. Immediately after injury, they undergo an activation event where they undergo a massive shape change. They spread extensively up to a couple of microns. They also take on this star-like shape. And they also begin to express surface receptors that allow them to interact with fibrin as well as other proteins at the wound site, so that way they can localize to the wound site and then also catalyze the clot formation. And so we a number of years ago, we started thinking about how can we break down platelet function at the very basic levels to basically mimic some of the key features that we were most interested in. And so in particular, we wanted something that could be injected, could go straight to sites of injury, like platelets do. So we wanted targeting. We also wanted augmentation of clot formation. And so we started, breaking it down. What are the minimum features that you can have in a material to mimic some of these functions? And so this is a summary of a lot of the things that that platelets do. So I already talked about activation. So we would want some sort of shape change more than likely. Platelets adhere to the injured vessel wall. They also aggregate. My group doesn't actually focus on that, but platelet aggregation is important. Facilitation of fibrin formation. And then again, platelets play a really large role in immune responses as well. And so we started thinking about how can we mimic some of these things. And the approach that we took is actually relatively simple, but as I'll show you throughout a couple of slides, despite the simplicity of this design, we're able to really mimic some of these key features of platelets. And so what we did is we took a polymeric microgel, so this is just a colloidal hydrogel that mimics the size and mechanical features of native platelets, and then we couple it to a nanobody or an antibody fragment that binds specifically to fibrin that's forming at the wound site. So I don't have this in my slide, but we heard earlier about this idea of using directed evolution in order to mimic biology. And that's actually something that we incorporated into this design as well. So we used molecular directed evolution, starting with a nanobody library in order to identify nanobodies that would allow us to target wound sites without interacting with fibrinogen, which is the precursor of fibrin that we were interested in targeting. So we used directed evolution to identify nanobodies that would only bind to the wound site without interacting with the precursor form of the molecule in the bloodstream. And so it turns out just by simply mimicking mechanics, shape, and then binding ability, we really are able to get some really fascinating features. And so for the last, really just the rest of my talk, I'm going to show examples of this technology in practice. And so some of the things that we found, breaking the design feature down into its minimal elements, we found that having replicating the ability of platelets to bind to fibrin allows us to induce clotting. And so we showed that in a number of in vitro studies. So these are, this is also using some biomimicry. We're actually testing in the lab using endothelialized microfluidics devices where we're mimicking microvasculature. And then here we're testing the ability of the particles to induce clotting in this vasculature on a chip model. And we see that if we have fibrin binding, we're able to induce clotting, but without that, we don't really see much clotting. We've done a substantial number of in vivo studies as well, investigating the ability of these particles to, number one, target to wound sites and then augment fibrin formation, clot formation. And this is some data from a rat trauma model where we see basically clot formation here in red at a wound site. And there's something weird going on with formatting here, but we also see co-localization of the synthetic platelets at those fibrin sites. And some other really key advantages or some of the really cool things that we found from this very simple design is not only is the targeting really important, but mimicking the mechanics and the shape of individual platelets also turns out to be really important for replicating certain platelet features. And so I mentioned earlier the particles that we're using in particular allow us to mimic the shape and mechanics of platelets. So these are some scanning electron microscopy images that show inactive platelets compared to active platelets, so you can really appreciate this massive change in shape. One thing that I haven't mentioned yet is the microgel particles that we use to create these synthetic platelets. We create them through a synthesis scheme where we don't add any exogenous cross-linker. And it turns out that these particles, they're really loosely cross-linked. And so that allows them to change shape and spread really extensively. So this is a picture of our particles, which we call ultra-low cross-linked. So you can see that they really mimic this activated platelet morphology in contrast to more traditionally cross-linked particles. And it turns out that ability to mimic platelet morphology allows us to also mimic something that other synthetic platelets had not previously mimicked, and that's this ability of platelets to retract clots. So I mentioned that on my first slide, but this is a feature or a mechanism by which platelets pull on the fibrin network and make it more stable over time. It also makes the clot smaller, which allows for blood flow to be restored. And it turns out that it's important for mechanical stimulation for wound healing events as well. So these are clots with our synthetic platelets, and these are clots with actual platelets. So you can see that we're able to recapitulate that. And so we've shown in a couple of studies that just having these very simple features of mimicking size, shape mechanics, and targeting ability allows us to induce clotting as well as promote healing. So these are some studies where we looked at that. So one of our hypotheses was that by having this clot retraction, we would get increased clot mechanics and drive durotactic mechanisms. And so these are some in vivo studies that show that if we apply the synthetic platelets to full thickness dermal injuries, we do in fact see enhanced healing. And then in vitro studies show that we're supporting cell migration into those PLP-laden clots as well. And I'm just going to run through the last couple of slides quickly to see if I can get us back on time. But we've really shown in a number of animal models that just these very simple designs and just picking key features of native platelets allows us to really see great functionality in vivo. And so these are some experiments where we've administered the particles in a wild type model in mice of traumatic injury. And we see a nice dose response with the particles where when they're delivered IV, prior or even after the injury, this particular experiment is prior to the injury, we see a significant decrease in blood loss. We also see, this is a longer term study in the same model, but we're looking 7 days out, and we see that the clot retraction feature of these particles allows for enhanced healing 7 days after that traumatic injury. We see really nice safety profiles of the particles as well. The softness of the particles and mimicking the mechanics of the platelets allows them to be cleared within about 24 hours or so. And we haven't seen any safety concerns so far. And then just lastly, we also, our hope is to eventually translate these synthetic platelets into actual clinical use. And of course, the step before clinical trials is testing these in large animal models. So in collaboration with Lauren Schnabel and her student Drew Koch at NC State CVM, we've been able to translate this to a large animal trauma model using pigs, which are the gold standard for testing hemostatic materials prior to clinical trials. And here what we've seen is in these large animal models, we're able to replicate what we've seen in the smaller models where our synthetic platelets are able to significantly decrease blood loss. One thing that I want to point out is actually in this case, our particles actually even perform better than platelets that we isolate from pigs. So here if we're able to use about 10-fold less compared to a normal clinical dose of what you would administer with platelets. So this is on a platelet to particle matched number basis, our particles perform much, much better. We've also just in the last minute or two here, we've looked at, we're in the process of looking at other applications of this. And this is an application in traumatic brain injury. And so again, these very simple designs allow us to address numerous biological problems. So we've seen that we can decrease blood-brain barrier permeability after TBI with these particles, and also decrease neural inflammation up to 7 days after TBI. So pretty excited about that. So some ongoing work, we're continuing to think about how can we add additional functionality, how can we better mimic platelet behavior and functions. And so just some examples, we've started thinking about mimicking platelet immune function. So these are places where we could really benefit from interactions with microbiologists, starting to think about maybe delivering novel antimicrobial agents with our particles, because they are great for drug delivery as well. We've started thinking about other regenerative medicine applications. Platelets are really, they've gained a lot of utility in regenerative medicine, but natural platelets have lots of downfalls, like needing donors and variability. And so we've started thinking about those types of applications. And then, so just to summarize and make sure that I address some of the key things that are important for this symposium. So just in summary, what we're really trying to do is mimic platelet function to stop bleeding and improve healing. The way that we started with this design was by looking at the literature, drawing inspiration from native platelet biology, working closely with platelet biologists and those that were really experts in hemostasis as well, to develop some of this initial design. The particular aspect of platelet features that we mimicked here were platelet morphology and fibrin binding. And those key features have really helped us achieve our engineering goals from the outset. And then just in the spirit of this symposium, some connections, I mentioned this on the last slide, but just to kind of summarize, some of the connections that I see as helping us further this work and potentially areas to bring even more biomimicry in would be working, even more closely with platelet biologists, clinicians, veterinarians, as well as with experts in scale-up and manufacturing, physicists, mathematicians. I think that there's a lot of opportunity in this particular example to connect with multiple interdisciplinary teams to continue to improve and iterate on these ideas of engineering cells for medical applications. Okay, and so I'll stop there. Just always want to thank lab funding sources and collaborators. Thank you guys for your attention, and I know we're about to go into a break, so happy to take questions during the break, I suppose. Thanks.
Thank you so much, Ashley. Thank you so much, Ashley, and thanks to all the first batch of speakers. Look for the speakers in the break. Wait for them to run to the bathroom, but if you want to follow up. Really an amazing diversity of approaches to biomimicry this morning, and I think one of our real opportunities and challenges is how do we connect across all of these areas. And so we'll take till 10:55 for a break, and then we'll reconnect around some lightning talks that bring even more perspectives on what's possible here. But thank you for an amazing first session, and look forward to what's to follow. We're going to go ahead and get started for this next session. John. So for this next session, we're going to have a series of eight-minute talks. And these will be talks that are sort of slated for about 5 minutes with questions. I suspect we'll, run toward a little long in 5 minutes, but Lauren Nichols is going to hold up a sign for speakers when you get to 5 minutes, just to keep us honest on time. And what we're going to do is run through the first 4 talks and take a little standing break for a second, and then we'll go through the next talks. And these are shorter talks to give us even more of a sense of the diversity of kinds of research in the realm of biomimicry. I think we already saw in the first session that biomimicry really covers this extraordinary expanse, from really organismal biology to tweaking individual molecules, to things at the interface of those disciplines, to work that really spends most of its time on the side of math or in engineering. And for me, that's the real opportunity, but it's also the challenge. How do we work across all of those fields? And so I think we'll see even more diversity this afternoon. Our first speaker this afternoon is going to be Natalie Levin, who is from the Department of Forest Biomaterials here at NC State. And her talk title is Bio-Inspired Textiles and Objects for Sustainable Fashion Displays. Thank you so much, Natalie.
Thank you. All right. Thank you so much for the introduction. Good morning, everyone. Happy to be the first to present a bit our work on bio-inspired textile and objects. So just to jump in the topic directly, we get our inspiration and raw materials from the biomass and mostly tree. We love trees, not only because we are the Department of Forest Biomaterials, but because they also have a great structure, as you can see here, a very oriented porous structure. They are a hierarchical structure with building blocks that we are using in our work for creating those beautiful iridescent color that we have discussed this morning again when it comes to the morpho butterfly here. So we use wood for its properties, its chemical composition, but also as a building block here, we have found a way to use wood at the nanoscale. And this is what I'm going to show you here, because those particles are the ones who are responsible for this beautiful iridescent color. So if you look more closely at the hierarchical structure of wood, well, researchers have found a long time ago that the building block of wood is actually some crystal phase called cellulose nanocrystal that really looks like those uncooked grain of rice, right? And those little crystals are made of the cellulose chains that are tied together to make this crystalline structure. And this is a type of particle I'm gonna discuss today. We also do a lot of work on another type of particles that really look like cooked spaghetti, but that will be for another time. So why are we interested into those particles? Well, we have found out that in water, they self-assemble into a specific way that interact with light, so that you can see those beautiful colors here. So I will not go through the physics behind the interaction of the particles with light, but the idea is that from a certain concentration in water, they self-assemble into these very nice DNA-like shapes. And if you control basically the space between their different planes, you can control their interaction with light. As a result, you can see those beautiful light just by visible eyes. So in our lab, we work a lot on those nanocrystals, and we try to control the colors because it's not that easy. Naturally, if you just dry them and remove water, you will get this beautiful blue. But how can we get to red, yellow, purple, right? And this is the whole challenge here, is controlling the color to a point that we can have an even color and distribution. So here, for instance, by playing with some chemistry, we can get a green-like color, an orange, yellow, and a red. But still, it's not that easy to maintain depending on the surface properties that you are trying to apply them on. So we are still working on that, and our main objectives here is, yes, to decipher the mechanism of self-assembly of those CNC particles to make sure that we can control the color formation and its evenness, right? And we currently have with Dr. Lucia and Lucia, a PhD student working actively on this fundamental topic. But we are also trying to understand how those particles will interact with different substrate. For instance, if you were to decorate textile materials or wood surfaces or metals, you can see here that if you were to use exactly the same formulation on a plastic substrate, which is here polypropylene, you will not get the same color at all on a textile product, simply because of differences in porosity, in surface chemistry, and so on. So here this example is quite basic. We can understand clearly the differences between plastic and textile, right? But you will also have differences between different types of plastic as well. So here we are trying to understand the influence of defect, of surface chemistry, porosity, and density of the substrate to understand how we can control the colors of the particles. So I'm going to show you a little video because what we started actually is not by the fundamental research, but we started actually by the prototyping of this product. So we really got inspired by those element of nature, the morpho butterfly, the jewels beetle, to get those vibrant colors. So those structural colors compared to dyes and pigment that we commonly use for textile coloration, they are long-lasting. They will not fade under sunlight. They are also non-polluting and non-toxic. This is a great appeal to the textile industry. So we can basically, from the biomass, extract the crystals, so those building blocks, and then make a suspension that really can be used like paint. Of course, it's a waterborne coating or waterborne painting. So here the whole challenge is how you control the evaporation of water and the amount of water as well, depending on your surface. But if here you can see that upon drying, this is a an accelerated video, you get those assembly of the particles into different colors. So we can create different colors depending on how we control the chemistry between two planes. And we also have scaled up the possibility of making customized shape and pattern with those crystals. We apply them directly on textile, and depending on the angle of where you look at basically the textile product, you will see different color. So this is, for instance, the different prototypes we built. We also got the chance to participate to a fashion show in Raleigh, where several of the mannequins, models were wearing our prototype of t-shirt here. But you see here that we can literally design the shape, the forms that we want. Again, the idea here to apply that on textile was quite challenging since the textile is a highly porous materials. So we had to work with another technology strategy here and work with films that would be like some foil that you would apply on different surfaces. Another project we are currently developing in collaboration with the College of Design is happening here on main campus. So we are redesigning the Biltmore courtyard space of our college. So the College of Architecture of Design with the architecture team, they build up those metallic structures that aims to mimic the bark and the wavy structure of bark on the wall here. And using our CNC coloration, we are gonna bring in some coloration here by applying those little strips of CNC that will also actually change color as a function of the humidity. Since it's all about their interaction with water, so if you had a very hot, humid weather, you will have a color shifting from blue to red. So this is a project that is still undergoing, since it will be the first time large-scale outdoor application of this material, so we still don't know how they are gonna react. So it was just a very brief introduction and overview of what we do with wood at the nanoscale to create sustainable coloration. Thank you.
We have time for one quick question.
Yeah.
Can you explain more about the colors being applied to the t-shirts, like through printing?
So what we do right now is like we create some films just by evaporation of the solvent, and we apply the films directly on textiles through a green chemistry pathway. I cannot give details because it's under licensing and now patenting. So we are working with industry to get this work further commercialized and advanced to apply that on different surfaces.
Thank you so much, Natalie.
Thank you.
So the next speaker is Corinne Zhao in the Department of Mechanical and Aerospace Engineering. And Corinne will talk about rational design of viral-inspired nanoparticles for cancer detection. Thank you, Corinne.
Thank you. Thank you for the introduction, and thank you for everyone being here. So I'm Corinne Zhao from Department of Mechanical Engineering, and I'm working with Dr. Jingjie Hu now. So today my topic is going to be the rational design of the virus-inspired nanoparticles of cancer detection. So just like the project it's inspired by the virus. Just like virus, they have the special ligands can be detected by the special receptors of human beings, so that they can get into human bodies, human beings' cell and get people sick. So the nanoparticles in this project is designed, decorated with the special ligands that can be detected by the receptor on the breast cancer cell. So with the enhanced MRI signal or other functionalized, so it can be seen under MRI and it can do the early cancer detection. So a lot of people, in fact, a lot of groups have done this process before, and we are developing a new material, of course, but what's really new, what's really fascinating in this research. So we are not from material science, we are not from bio, we from mechanical engineering, and what we care about is the force. We care about the adhesion, we care about the energy. So what is the adhesion here? We have a cell, and we have a lot of particles. So when the particles get around the cell, how easy are they going to be absorbed onto the surface of the cell? And that will further determine how many particles are going into the cell. And also about the endocytosis, this is basically the entry of nanoparticles. So we are studying what kind of particles will be in the cells more than the others. So for this part, to get the adhesion force, we started from experimental part by the AFM as a regular method, and also we did the calculation for the molecular dynamic simulation. So this can not only describe what's the process of the particles attach and going into the cell, it can also serve as a predictor, what will those particles we want to design. And by the enter part of the particles into the cell, so the thermodynamics and the kinetics modeling was done here. This can also serve as to enhance the cellular uptake and help us design the nanoparticles. So when the nanoparticles are entering into the cells, there will be a change in the membrane, the free energy change in the membrane. So with those models we developed, we can predict the range of the cluster sizes that can enter into the cells, and we can estimate the wrapping time, and we can get the optimal particle size. So these things will further help us in the application, so that we can have the bag particles for drug delivery, for imaging, or for something like hyperthermia. So this is our group. We are a very new group with Dr. Jingjie Hu, and that's all for my presentation. I would like to thank you all for being here listening, and also I would like to appreciate our supporters. That's where my salaries come from, right? So thank you all.
Time for questions, if we have any. If not, we can, thank you again, Corinne. If we can welcome Tom Schrader and, oh, I just lost your title. Tom, I will go back quickly and find it.
Let's see.
Do you have it up there?
One sec. Which of these clickers works, I wonder? I can just stand here. That's cool.
What do we got? So Tom is from the Department of Textile Engineering, Chemistry, and Science. We're going to be talking about the fabrication schemes for crystalline materials inspired by biomineralization processes. And I came to know Tom's work through a totally different project, reimagining how we think about electricity in animals. And so I think there are lots of potential projects here. So thank you so much, Tom.
Thank you. Yeah, and I am going to touch on that real quick. But hello, everyone. My name is Tom Schrader. I'm a new professor in the College of Textiles. My background is chemical engineering and polymer science, but I hope to kind of apply it to that context. And so I only just arrived in the fall, which means that what I'm going to talk to you about today is more like future plans than stuff I have actually done already. But first, I do want to touch on kind of the process of biomimicry as it happens in my group. And I do have a confession, which is that up till now, I have been one of those biomimicry business as usual engineers who opportunistically combs the literature for interesting sort of systems thus far and have not directly involved biologists so much in my work. That's something that I hope to, fix in the future. But so essentially what this has looked like is to, identify some biological organism or process where there's an adaptation functionally in nature that we, human engineers could benefit from potentially. So then an example of this is the electric eel, which during my PhD, we sort of became fascinated by as a source of electricity that is generated under metabolic biological constraints, essentially with using energy that comes from food. Generates these huge voltages, which is, a remarkable feat, and it's the reason they're famous. So then kind of the second step is to develop an understanding of the mechanism behind this adaptation. And so for us, we essentially recognize that the 80% of the anatomy of the eel is composed of sort of this set of 4 repeating units, 2 membranes and two reservoirs containing different salt concentrations with different ionic selectivity. And sort of those diffusive driving forces are what ultimately kind of generates the shocks of the eel, and that we could potentially recapitulate this in systems using sort of salty reservoirs and selective membranes ourselves. And then kind of the third step is to design an engineered system that uses the same mechanisms as what we have observed in nature, or what biologists before us have observed in nature. And for us, that was to develop a series of hydrogels that, again, kind of recapitulated these 2 sort of reservoir function and 2 sort of membrane function elements, put them together in series and generate voltages and electrical current as a result. And we found ourselves able to actually kind of power systems in this way using this type of architecture and bio-derived mechanism. So now I'm here in a new context in a new university, and starting up a research group where at least one of the focal areas is going to be the interactions between polymers and crystals. So nature is, of course, a great showcase of using sort of macromolecules to control crystallization processes. Probably the best sort of family of examples of this is in the context of skeletal structures like bones and sea urchin spines, for example, in the ocean. So both of these both use a scaffold of collagen or chitin or some other biomacromolecule, but also use process directing agents that are macromolecular as well, in order to generate these fantastically robust and very intricately structured elements. Another example is antifreeze proteins and ice nucleating proteins that are expressed in various organisms that grow in places where the temperature dips below the freezing point of water. Essentially, these are proteins that are capable of binding to the surface of ice crystals or serve as nucleating points for those ice crystals, and essentially are able to manipulate crystallization in this way. So there's kind of 2 modes that they can serve, one an inhibitory function for crystal growth and one a kind of nucleating function for crystal growth. And the reason that we human engineers might care about this is that crystals are kind of everywhere. They're useful for a whole bunch of different purposes, and they have really unique properties, optical properties, mechanical properties, and electrical properties as well. They touch a lot of kind of different areas of life and are at the core of a lot of the advanced materials that we use in life now. And also, polymers are already being used to control crystal growth. An example is quantum dots. Quantum dots, which are used in biomedical imaging as well as contemporary like TV displays, basically, are often grown in a very controlled manner using capping ligands, many of which are polymeric in nature. This is a relatively simple control scheme compared to what happens in biology, but it's still an example of this process at play. So a lot of what I want to work on in my group has to do with this hypothesis of how bones form. And essentially this hypothesis is called the polymer-induced liquid precursor process. It involves the deposition of salts, usually calcium salts, on biopolymers such as collagen and chitin. And essentially, you require both those two ingredients, the salts and the scaffold, but then also a sort of non-collagenous protein process directing unit, which is usually a polyelectrolyte to direct deposition. So without that process directing group, you wind up, when you put an insoluble salt together on a scaffold like this with a mess, pretty much. With that process directing polyelectrolyte, you get sort of this intrafibrillar mineralization process that happens very nicely. The crystals are aligned, which shows up in scattering. And this produces kind of the robust biominerals that we see in nature. So essentially, there have been, for the past 20 or so years, some people who have experimented with this in vitro. This involves essentially the non-collagenous protein that is directing this, directs a liquid-liquid phase separation process that allows the mineral to exist in sort of a liquid precursor phase that can then infiltrate the sort of biomolecular scaffold. So people have made all sorts of fancy geometries of simple salts in this way, like inverse opals and gyroids. What I want to do is, I guess, essentially more mechanistic study of how this happens in biology and also in vitro context, and then to take it outside the biological context to synthesize sort of advanced crystalline materials potentially beyond simple salts, like metal organic frameworks, zeolites, etc., but then also to use kind of those simple salts as mechanical reinforcement for things like textiles. So that is what I plan to do. I am new in town and willing to collaborate, very eager to collaborate, in fact. And so I encourage y'all to check out my website and approach me after the talk.
Thank you so much, Tom. Mm-hmm. We have time for one question, if there's a question. All right, we'll keep moving, and you can circle back with Tom. Thank you so much. So our next speaker is Rohan Shirwalkar, who's the professor in the Edward Fitz Department of Industrial and Systems Engineering. And his talk will be ultrasound-assisted bioprinting to achieve biomimetic organization in engineered tissues. Thank you so much.
Thank you, Rob. And I simplified the title to really help everybody appreciate the core topic.
Not in time to help me.
All right, it's a pleasure being here. And in the next 5 minutes, I hope to highlight to you some complementary approaches, but also appreciate the role manufacturing processes can play in achieving biomimicry. So this is not so much about studying biomimicry at its fundamental level, but a more top-down approach on how you can make it. And my talk today is really going to deal with research in making tissues and organs. So let me start with this. I work in the orthopedic domain and always like to highlight the knee joint as a source of pain, because either every person in this room themselves or their family or friends have experienced or continue to experience knee joint disorders, right? Whether it be a sports injury, whether it be osteoarthritis, and there's a lot of different things that can go wrong. In today's day and age, the solutions for these really include metal implants or grafts. Honestly, this is basically like using duct tape. I'm very generalizing it, but that's what it is, where these are not optimal solutions for good longer-term outcomes. They can work for a few years. In fact, when grafts are put, they are put in younger patients with the knowledge that they will only prolong when they are going to need a metal implant. So better solutions and approaches to this, and in the context of a biomimicry symposium, is being able to make tissues and organs using living cells, biomaterials, and the right type of molecular cues there, the growth factors. And really, these would be better longer-term solutions to what we have today. In making these, however, I think while this 3D shape and geometry is very important, and we can appreciate that just in our day-to-day lives when we see it, what's also important is what's underneath. So if you look at it more microscopically, any tissue in our body, there is a very unique organizations of cells and the proteins in there. And this plays a role in their biomechanical function as well. And so when we want to make tissues which are really functional, it's not only capturing the macro structure of it, but also the microstructure of it effectively. And that is where there's a lot of room for continued work, and I certainly work in that. So, through several collaborations, for example, with faculty, like Dr. Matt Fisher in biomedical engineering or Dr. Benham Podihemi in the College of Textiles, we are investigating a variety of processes because really to the spirit that Rob highlighted earlier, we have to go in thinking we don't know the final solution and really explore and try to understand. And so we are looking at a variety of processes trying to figure out which one might get us the best biomimicry in these. And let me highlight one example of a process that we recently developed. It's a patent pending process where we are using a combination of 3D printing and acoustics ultrasound to achieve biomimicry in cells and the proteins they form. And eventually they will lead to the functionality that we look for. So in this process, what happens at the macro level is you are using one of the more standardized 3D printing processes like extrusion, or this is a laser-based curing, this is a photosensitive material. But at the micro level, while this printing is happening, we apply ultrasound waves in a very particular way. And I'll show you a video here. In about 10 seconds, you will see a lot of these pink dots that are cells that organize in very specific patterns to replicate things that we see in ligaments, tendons, menisci. And so this is a real-time application of ultrasound to that process, and you'll see horizontally how the cells have started aligning. And we essentially can create a variety of distinct patterns. We can also, I showed you just a snapshot, but really we can create patterns relevant to many tissues, like for the knee meniscus, we can create circumferential patterns. It's not just achieving that initial alignment of cells, but through this and other processes, we also ensure that the proteins and the extracellular matrix that then the cells produce over time is also aligned very specifically to mimic what we see in our tissues and such. And a concluding thought. So I talked about a knee joint tissue, but really this concept applies to many different applications. One of the ones we are very recently gotten involved in is creating cultivated meat. And so this is not plant-based meat substitutes, but this is how do we take porcine and bovine cells and grow meat that tastes like real meat without sacrificing an animal. So these kinds of processes and concepts do apply to that as well. So with that, I'll conclude, acknowledge some of the grants and students that worked on this project, and happy to take questions.
Thank you so much, Rohan. Any questions?
I just wondered, for the knee replacements, is there somebody actually working on applying that, and it will be available soon somewhere
Asking for a friend. Yeah.
And I'll tell you this, at every seminar I give, this is always a question. Like this is a constant question. So the short answer is no, in that it's going to take time, because even while we are achieving this biomimicry, there's so much more to do even at the molecular level. And it really has to be the convergence of approaches, and that is still going to take time. So we can make some things here and there, maybe bone replacement, but really to achieve this meniscus ligaments, it's going to be a few years. Okay. Yeah.
Well, thank you so much, Rohan. And we'll applaud this first group, and again, and we'll take a little 5 or 10 minute break just to stand up. I know we've been doing this for a while, and then we'll resume with the next 4 talks. my mic on? Hey folks, we're going to get started again. We have just 4 more talks. And I'm super energized by all these side conversations that are happening. And so, as are the side conversations. So one thing I'll say is, I mean, that I really hope what this engenders is that, whatever product comes out of this afternoon and whatever conversations we have today, but also new ways of continuing to talk about these topics. I mean, they cover extraordinary terrain. And so if you want to continue to be involved in these conversations, let me know. If you have ideas about ways we can facilitate some of this work, let me know. I mean, I was trying to think, like, could we actually, pose a molecular challenge, pose an engineering challenge, and then, could my office help search out the biologists that might bring the comparative context to that challenge? Could we do something like that? But I suspect we have all sorts of ideas bubbling up in this room. And so please don't hesitate to share those ideas, because this is new, but we're really pulling in this room people from many departments and backgrounds. And so I think the potential here is great. And so don't hesitate to reach out. So the first talk of this last section is by Caroline Prue. And Caroline is in the Department of Chemistry, and she'll be talking about new strategies for peptide mimicry. Thank you so much, Caroline.
All right, thank you so much. And yeah, this is going to be very much molecular biomimicry. And so what I'm going to talk about is essentially trying to mimic the function of peptides, the bioactivity of peptides, and doing some structural modifications so we can address some of their drawbacks. And so one of the reasons why we're so interested in peptides and drug discovery is because they occupy really a unique space somewhere in between small molecules and larger biologics. And so that means they have some unique potential to target things like protein interfaces, for example. But of course, there are some drawbacks with using peptides in drug discovery. For example, you're using natural amino acid building blocks. And so that means that you will have usually poor stability, protease stability, and things like that. So we need to address that. But of course, again, not only can we target larger surface areas if you're looking at peptides for drug discovery, you tend to also have other advantages such as high potency, high selectivity, low toxicity, and so on and so forth. So we want to take advantage of that, but address the disadvantages of peptide-based drugs here. Okay, so in my lab, we think about how can we develop new tools, new chemical tools for the most part, to improve the therapeutic potential of peptides. And so we call that field peptidomimetics. We call these types of molecules peptidomimetics. All right, so very broadly, what are our goals? I've already mentioned that. We try to increase the stability of peptides. We would like to ideally, if we think about an unnatural amino acid building blocks, we would like to ideally be able to mimic all of the side chains that you see in natural amino acids. So I show some examples here: aliphatic, acidic, aromatic side chains, all of these different types of side chains, but using an unnatural amino acid building block. One thing that we think a lot about, so beyond stability, beyond amino acid side chain diversity, we also think a lot about conformation, because peptides, when they bind to their receptor or their other targets, really need to adopt a very specific conformation. So that's very important as well. A simple thing that you can do as a chemist is just to cyclize a linear peptide, and already by doing that, you can restrict the conformational freedom of your peptide. But more specifically, can we look at acyclic peptidomimetics that adopt very specific secondary structures, such as alpha helices, beta turns, and beta sheet type of secondary structures? So we would like to do that as well, beyond just cyclization. And then the third little thing that we think a lot about is synthetic E's, because we don't want to be the only one being able to do this chemistry in our labs. We want biochemists to be able to do that. We want biologists to be able to do that. And so we think a lot about solid phase peptide chemistry, and can we do the same type of chemistry, but to build our unnatural amino acid building blocks, essentially. Okay. All right, and then the last little thing, I guess, is late stage, a concept that we call late stage functionalization. So can I build the entire peptidomimetic and then introduce my side chain as a last step? That would allow you to construct a lot more libraries a lot more quickly. All right, so I just want to give you a very brief overview of what are these types of peptidomimetics. So there are lots of different ways that you can modify a backbone. One of the ways that we think about a lot is to do a single atom substitution here. So if you simply replace one carbon in a peptide, in a single amino acid in a peptide, by a nitrogen, if you're able to do that, chemically do that, there are actually a lot of consequences and beneficial consequences to doing that. So the first one is we're actually going to alter the hydrogen bonding, or also going to be affecting the conformation, which is something I talked about in my previous slide. So if you have now two adjacent nitrogen atoms, each of them has a lone pair. Lone pairs want to be 90 degrees from one another. So just by doing this single atom substitution, we are starting to reinforce beta turn secondary structures. We are losing the alpha chiral center. What does that mean? It actually means that we no longer have an L or D defined chiral center, but we can have adaptive chirality at that nitrogen now. But we are not changing where the side chain is, right? So the side chain exactly positionally between the nitrogen and the carbonyl group here. And overall, we are improving stability, not only towards proteolysis, but also chemical stability here. All right, so at the bottom of the slide here, you can just see the conformational properties of these ASA amino acids here. And so you can see that they do adopt this 90-degree angle between the two nitrogen atoms, which tends to translate overall into beta turn type of secondary structure. So you can see that a few examples here at the bottom of the slide adopting these turn-like geometries into crystal structures here. So just very briefly, again, we're doing, again, systematic single atom substitution here. In the first example, looking at azopeptides, we're maintaining the position of the side chain exactly at the same place where there used to be the carbon. But what about moving the side chain one on the other nitrogen, right? These are actually called azopeptoid. And what about having very highly functionalized di-functionalized azopeptides? Those are all things we can access in my lab now. And so very briefly, I just want to show you an example of how that has actually impacted receptor selectivity and bioactivity. And that's an example not in my lab, but in someone else's lab. So you can see not only having the carbon to nitrogen substitution, but also the position of the substituent can truly have an impact on biology here. So I'm going to move through just very, very quickly another type of peptidomimetic that we are interested in. We and others, I should say, is called a peptoid. And these are peptidomimetic where you move the side chain from the alpha carbon onto the nitrogen, completely different sets of consequence here. Now you no longer have a chiral center again at all, altered hydrogen bonding properties as well, but now you have no longer an NH hydrogen bond donor here. You have improved stability as well, but in this case, you actually increase the conformational freedom because you have a tertiary amide bond here. They are very, very easy to synthesize. You can actually make a 50-mer that has all unnatural and substituted glycine building block, so they can also be used as synthetic polymer mimetic. So just a couple of examples at the bottom of this slide here. We can use those in nanomaterials, antimicrobial peptoids, and also pretty good as polymer mimetics as well, which is something we've done. So I don't want to, again, take too long here. These are example of structure-inducing peptoid monomers. Again, one of the major drawbacks of peptoids is you're increasing stability. So if you want to use them as peptide mimetic, you actually have to start thinking about designing very specific side chains that will actually allow you to regain the rigidity that you want in these types of, so to speak, a little bit more floppy peptidomimetics or unsubstituted glycines. All right, so with that, since I really don't have a lot of time, last slide just shows you a couple of examples of an all unnatural peptoids that are capable of adopting well-defined secondary structures. We see examples of unsubstituted glycine oligomers adopting helical conformation, sheet conformations, and even unnatural types of secondary structure. So I'm going to leave it at that and open it for questions.
Thank you so much, Caroline. Any questions? Well, thank you. So our next speaker today is Lucian Lucia. And Lucian will be talking, is in the Department of Forest Biomaterials, and his talk is a bird nest bio-inspired strategy deployed for inducing biopolymer gelation. Gelation? You have to tell me.
Gelation.
Gelation.
Or you could say gelation. So it's a pleasure to be here. Thank you for your time. So this talk is, again, following on the piggybacking on a lot of, or birdie backing, I should say, on the backs of a lot of the talks here. We're going to talk about swiftlets, how they relate really to what we do in our lab. In our lab, as been said before, we do sustainable or green chemistries. Oh, I should grab the mic. I'm sorry. Okay. All right. Yeah, I'm sorry about that. So we do sustainable or green chemistry in the lab, and
we'd love to work with you. We worked with several of you folks before. And in this talk, what we're going to do is show how nature itself provides us means with creating new architectures, new motifs, so to speak, that we can use in society. And we take our inspiration from the swiftlets. These are species of bird. There's about 30 of them in the world. They more or less reside in Borneo, Malaysia, southern Pacific, northeastern Australia, a very interesting set of birds that make something called a bird nest that looks like an islet castle. It's really very, very cool. These birds, they actually settle on the side of cliffs to avoid getting eaten. And I don't know how they actually get on the side of a cliff and start that very first, structure, because myself, I can't even put a painting on the wall without falling, right? So these guys are on cliffs and they're putting their structures on. So it's a very cool process. And it's actually the males. So finally in the animal kingdom, we got the males doing a lot of the work. They spend 35 days setting up a home with their saliva, right? So we heard a talk earlier by David on wombat poop and whatnot, urination. We're gonna talk about saliva. So this is all very entertaining for us boys. We love all this nasty, crazy stuff. Okay, anyway, so it's actually made out of water, it's soluble protein, carbohydrates, iron, salt, and the rest is twigs. Can you guess what people do with this? Anybody? They eat it. Yes, have you tried? No, okay. Supposedly it's very delicious. It's a delicacy. It's bird nest soup, right? So folks that are from that part of the world, they will eat this. I know there's some people, this is great. I know, I'm so sorry, Tova. I know that you're just shrinking. Anyway, so the biomimicry concept here was trying to understand, how these guys use their saliva with twigs to make these beautiful biocomposites that could potentially give us a leg up, so to speak, or a wing up, right, on new structures that could be useful in society. And we decided, why don't we use what they use, but at the molecular level, right? So Natalie talked about cellulose nanocrystals, and we're going to take actually the cellulose itself with something that we will call saliva. And in this case, the saliva is this stuff right here. It's a derivative of cellulose called carboxymethyl, which is used in a lot of drug formulation, excipient formulations. It's something that we use quite a bit in society. And this actually is very sticky when it's in the protonated form, because this is polyanion by itself, right? It's got a lot of charge on it that basically resides in the backbone of this stuff, cellulose, which is found everywhere, probably the world's most dominant terraformer. Right, so we are taking this instead of dissolving, because typically what we would do as green chemists is we would try to figure out how to dissolve this in a green solvent. But what we've come up with, which is really, really, really clever, is to do this under a process which allows us to not dissolve it, but also, modify it as well, which is really very, very unique and very different. So what we've done, to make a long story short, is we've taken advantage of freeze thawing. We've been able to put together these systems, right? So I show you here the control, which is basically just the viscous CMC, carboxymethyl cellulose. And then we've added 3 parts to 1 of the cellulose to the saliva. So the twig to the saliva, and then the twig to saliva, 1-to-1 by mass. And what we've done then is we've gone ahead and taken them to the freeze thawing process, which then allows us to suspend them in an icicle formation, which for some of you, you may know that ice is such an incredible medium to work with because it can actually help orient things. And that's what basically we've been able to do, is to help orient them. And what I show you here are some SEMs in which you can see this bird net structure motif is actually working to interlock the cellulosic chains that basically consist of the backbone, right? And what we've been able to do is we've been able to do some rheology, very simple frequency sweeps, to get a feeling for how mechanically robust these are. And they're basically viscoelastic, they're rubbery, which is not surprising. The cellulose itself allows this material to be spongy and basically follows the trend of a, as you fortify it, as you percolate the cellulose throughout the structure, you get a much more robust, more of a quasi-solid that is represented here in these storage moduli curves all the way from H31 down to the control. And when we subject it to a creep, a compliance, that is when we put some force on it, so the bird is landing in its nest, and then it takes off the nest, how does that respond, right? Does it break apart? Does it respond in a sort of elastic modality? And it does, more or less. And it's very much in keeping with the backbone as the structural element to keeping this thing, suspended on the cliffside. And then, of course, we want to see how this thing gives us perhaps other properties. And one of the other properties that we fortuitously or serendipitously found was its omniphilicity. Again, not super surprising, because when I show you the mechanism for how it's formed, you'll get a feeling for this. So what we did here is essentially we tried to take a series of solvents from very low polarity to middle polarity all the way to the normal polarity of water. And what we see is that basically we're able to get absorption that's quite impressive with both of these structures. For the, this one should be the H31, my apologies. That structure is able to absorb quite a bit of DMSO relative also to the other structure. So, and we've been able to do this with bamboo fibers. So these hydrocolloids, these hydrogels, whatever you want to call them, are quite interesting. And we've done a lot of other tests that I'm not able to show you at this time. But the mechanism basically really relies on this freeze thawing process, where you're able to take the saliva represented by these little gumlets, right? And you have it sort of like satellites suspending around the cellulose. And then essentially when you freeze it, they sort of come together through the process of the ice sort of water causing these things to orient in this more or less crystalline pattern. And then when you thaw it, it kind of expands. Right, so you've got this very porous structure. We actually have made aerogels, foams out of it. So it's a really, really neat kind of process. Special thanks to the student that did this. And then thank you for your time and attention. I appreciate it.
Thank you so much, Lucian. We have time for a quick question, if there's a question.
I don't have any bird nest soup for you.
Yeah, great talk. Question is this: am I right that you would have to pretty carefully control the freezing rate in order to get the right, I guess, behavior between the front?
Yes. Actually, it's funny you mention that. When we've done freeze thawing a lot of times, and that actually is the case. We actually have to be sure that we do this in a very controlled manner, so everything is able. Yeah, because if you don't do it controlled, then it's completely amorphous.
Right.
Okay. Great comment. Thank you, Tom.
Thank you so much, Lucian. So first, I just want to remind speakers that you're all welcome to the working group this afternoon. So when we'll be working on pulling together these ideas, and if people have particular ideas they'd like to share for this afternoon who are not speakers, please reach out so we can get those into that conversation then. Our second to last talk is now from Tom Mikras in the Department of Molecular and Structural Biochemistry. And Tom's title is Enzymatic Carbon-Carbon Cleavage for Natural Product Biosynthesis. Tom?
All right, well, thank you very much for the kind introduction. So I thought I would give you the 10,000-foot kind of aerial view of what my lab is interested in, and then maybe if I have time, I'll do a concrete example. So we're interested in enzyme chemistry. All right. And ultimately, what we're really fascinated by is exploiting enzymes to make natural products of high value. And that can be either from the kind of biomass to biofuel kind of arena, how pathogens will make essential cofactors, and then these really kind of gorgeous antibiotic structures that, as Josh alluded to, can sometimes synthetically be really challenging to approach. Enzymes have figured out really elegant mechanisms to do it. So we think about how enzymes essentially tune for function. And I've been interested a lot in metalloenzymes that do some of the most difficult transformations known in nature. And if we think about it from a structural view or from an inorganic chemistry point of view, we always think that catalytic function is probably tuned by the immediate environment around the metal. And I've cherry-picked examples of going from something, a really simple biochemical function like carrying and releasing O2 to something like cleaving strong CH bonds. And we start making these correlations. Well, if I change the ligands in such a way, in this case, if I change the number of carboxylates, I can achieve stronger function. And essentially the idea is just charge neutralization. And I've cherry-picked this example extremely because it doesn't work at all. In fact, you will find all nitrogen ligands can do some really, really difficult chemistry. So it's important to make these structure function correlations when thinking about catalyst design. So in doing that, we thought, okay, well, a natural place where we could do this. This is a emerging new family of enzymes that we and other chemical biologists have been interested in called the HDOs. The ligation motif is exactly the same in all of them, but they do these varying different chemistries. And so now this has become an interesting playground for us to decide, all right, well, how do we get more sophisticated models for structure function correlation? All right, so this is what we've been doing for a few years in this family. This is the enzymologist form of the ground beetles. So this is the number of different proteins that we have at our disposal. There's more than 10,000 orthologs. Obviously, the emergence of being able to get sequence information so quickly has allowed us to identify a lot of them. What you see in the triangles is what's been characterized or what's been annotated at a chemical level. So there's a real need to understanding this biochemical world in much greater detail. And this will keep us busy for quite some time. All right, so I am going to show you kind of one example of mechanistic dissection and why that may be important. So, so this is folate at the top. You and I, every organism basically needs it for DNA synthesis. The way that most bacteria will make folate is through this multi-step pathway with charismate as a precursor. So chlamydia are kind of funny, right? They're intracellular bacteria. They've jettisoned most of their biosynthetic pathways because they do have this parasitic lifestyle, but they have this kind of weird way of making folate. So this uses a dimetal enzyme called CADD. And so to take a very long story short, this enzyme, and what we've learned from this enzyme is here's the dimetal site at the bottom. It will actually use its own tyrosine, cleave it off, process it, make that central portion of folate. So the enzyme, so this is quite a weird example because it's teaching us how one enzyme can sandwich lots and lots of different activities. And in a way, this is really, I think, instructive for catalyst design. We can do long-range chemistry. We can also do close-range chemistry. It can do amination. It can do a lot of things all within one protein scaffold. So this is kind of one example that we think of biomimicry, or at least in terms of learning from enzymology, learning from protein biochemistry, to start thinking about what kind of lessons for catalyst design that we can develop. So thank you so much.
Stephanie's joining us from Campbell University from the biology department. And Stephanie's going to be talking about biomimicry and camel cricket microbes. And Stephanie will talk about still another dimension of biomimicry that we can think about as biomimicry or not, where one actually borrows from another organism. And so rather than mimicking, it sort of we steal it. And how do we think about that? And so thank you, Stephanie. Yeah.
All right, so today I'm going to start out by introducing the term xylophagy, which is a really fancy term just to refer to an organism that might eat and digest wood. So when you think about this, what organisms do you think of? Oh, maybe not working. Termites. Oh, no. Great job. Okay, termites. Fantastic job. A+. Some of you said wood louse, or might have been thinking. And then we move next to beetles. I heard beetles back there. Great job. You may even think of shipworms. Oh, this is, thank you. Fantastic. You may even think of shipworms. Again, we see our beetles. And then there could even be, there's an article about even catfish from the Amazon that eat wood, right? So we have a lot of really interesting organisms. But what if there were organisms participating in xylophagy that are a little bit more commonly around you? So we think about termites and how we don't want them in our homes. But what about arthropods that may live in your house that potentially participate in xylophagy? And so from some work done at NC State with Matt Bertone and others, they looked at all of the arthropods inside your house. And so you may see some of these and think, okay, flies, spiders, some other things. But what we really want to focus on, or what I want to focus on today, is the camel cricket. And so I'll be talking to you about biomimicry in the camel cricket microbiome. Okay. So the camel cricket, it's pretty common in North America. Have any of you seen camel crickets in your basements or crawl spaces? Most of them are like, there's some like ews, right? Because they're kind of creepy and they're blind. So when they jump and they jump on you, they get even creepier, right? Oh, yeah, sorry, hooray. So there are about 150 different species in North America alone, so there are a lot of them. And as we look at the natural history of the camel cricket, it's really interesting that they are a keystone species in nutrient-limited environments, especially in cave systems. And so we want to think about how they make a living in caves or low-nutrient environments. And so this is where the termite comes back into play, right? So both of these organisms are making a living by digesting the material that makes up wood. And what we want to figure out is exactly how they're overlapping. So we know a lot about the termite in terms of xylophagy. There are a lot of enzymes that the termite makes, as well as the microbiome in the termite that's allowing it to digest the woody material. So this is just a very short list of some of the microbes in the termite gut that's allowing it to break down wood. From the limited work that I've done, looking at just a couple of camel crickets, really not that many, we have cultured, not sequenced, a small list of organisms. And if you look closely, there's not much overlap. So how are they doing this, right? They're both participating in a process that's kind of hard to do to break down wood. And they're probably using bacteria that's living in their guts, as well as the enzymes they produce to break this down. And so we're trying to figure out how it does this. Okay, back to wood. So we know that the termite breaks down woody material. We know that the camel cricket is probably doing this as well, and we're not sure how. When we think about the structure of wood, thank you to those who've already talked about wood structure. We know that there is cellulose, hemicellulose. These are sugar molecules. They're a bit easier to break down, so we have an idea of how the termite is doing this pretty well. But the other remaining item that no one likes to talk about is lignin. And so lignin is really hard to break down, and so we want to figure out how the termite, especially the camel cricket, living in really low nutrient environments, is breaking down not only cellulose, hemicellulose, but also lignin. So with this work, we looked at bacteria that we could culture from the camel cricket. You see on this list here, and then different substrates that mimic components of wood or woody material itself. Now there's one bacterium that we cultured that's especially interesting because it can grow on most of the things that we gave it to grow on that resembles wood or a component of wood. And then I want to draw your attention even further with the things that contain mostly lignin. So if we look at industrial waste that contains mostly lignin, or even lignin itself, we're finding that this bacterium can grow on lignin as its sole carbon source, suggesting that it's probably breaking it down in some way, shape, or form. Now, I've talked about lignin, but I haven't shown you its structure. And I love to show the structure because you don't have to know much about chemistry just to be like, oh, gross, right? Or at least maybe that's when you see it. And so we're trying to think more about how the camel cricket breaks down lignin because it's a really complicated process. And so the microorganisms within the camel cricket and likely in the termite are contributing in some way, shape, or form to lignin degradation. We took this a little bit further with the camel cricket, and we looked at its ability to process a substrate similar to lignin, ABTS, showing that it actually has this activity. And we see it around pH 6 when we grow that organism from the camel cricket on lignin-containing compounds. So this nice tall orange bar here. So this is a very interesting environment where we see biomimicry of the breakdown of wood. We know that the termite has a lot of bacteria or yeast that are contributing to this process, and we know a little bit about the camel cricket and the microorganisms and enzymes that it might be using as well. However, we've just scratched the surface. So there's a lot more work to come, not only in microbiology, but also with ecologists thinking about where organisms live in strange environments. Then we can partner with anthropologists, entomologists to think about other things that are happening, other organisms that participate in this process, microbiologists who can potentially help culture those things, and then biochemists so we can look at the enzymatic process that's carrying this out. Okay, thank you so much. Any questions, or are we finished?
Sure.
Very nice talk. We were wondering, did you characterize by any chance the lignin, the quality of the lignin? Because we were wondering if it potentially could be useful for some end application.
So after degradation, the characteristics of lignin, I've done that with some other very briefly with some other organisms, and we know that some bacteria that break down lignin can actually produce valuable byproducts, and so that would be the overall goal, is taking lignin, which is typically treated as waste, and then converting it into something that's more valuable, and using the microorganisms from camel crickets to kind of inform that work, right? And so instead of treating lignin as something you extract and burn, something that you extract and utilize for value. Yeah.
Thank you so much, Stephanie. Well, so that's our last talk for today. Let's thank all the speakers again. It's really been an amazing collection of talks. We're going to break for a while, and then the speakers can convene in a working group down the hall here a little bit later, which I really look forward to. Does anybody have any sort of overall questions, or questions they didn't get to ask earlier while we're still gathered here, or is everybody ready to go for lunch? Yeah.
Already pointed it. I think it's very good to include this in the beginning of the work. Even big pharma now in Europe, they need to do it right away when they are developing new drugs. So no matter what you are going to develop, it's very nice to think about soil, aquatic, and there are some nice tests that can be done, like at least to address this and think about that. So the cycle really should include that, in my opinion.
I think that's a great point, and Heather Patisal was making that similar point earlier, and I think we have the people here to better make those connections, and so I mean, there's some exciting conversations that we can still have. Yeah, a great point. Thank you. Anybody else? Well, thank you all. I'll stick around a little bit if there's follow-up, and then the speakers can convene down the hall for some lunch in a little bit. Thanks again.